identification and validation of novel therapeutic targets, significantly contributed within primary and secondary screening of NCEs for a variety of therapeutic targets, including enzymes, proteins, particularly in the area of central nervous system, cardiovascular system, diabetes, obesity, and safety and toxicology aspects. Currently actively involved in DBT, Ayush, and industry sponsored projects. Now I take the privilege to invite him for sharing his thoughtful and fruitful words.
Thank you very much. Happy morning to one and all. Respected Silvamurthy sir, Honorable Minister, other dignitaries present in the summit, participants, delegates of National Summit 2020 Innovations in Biotechnology Era. It is an absolute honor to be here and to among you share my viewpoint and the importance of this important summit. The National Summit Innovation Biotechnology Era is inspired by idea of United Nations Development Program of Sustainable Development Goals 2030 that emphasize the idea of continuous contributions and innovations. Other than that, it is also emphasized from the idea of our Honorable Prime Minister Sri Narendra Modi ji's 8-point vision for the 21st century. India and the unite the entire country by adopting good governance and using the technology that we are using today. The National Summit 2020 would help on a massive scale, which would benefit the young minds in several ways. The summit would provide an opportunity for research interaction, privilege for young researchers to learn and expand knowledge, professional speakers, academicians, practitioners, and government officials for sharing their ideas pertaining to applications of biotechnology research and innovation. Briefly telling about the JAX Foundation, which is organizing the event. It is an international organization supported by BRICS International Forum and recognized by Government of India under the Indian Trust Act 1882. It is working for the society, for the welfare of young generation and promoting social or political change and play a critical role in developing society, improving communities and promoting citizens participating for the fulfilling the dreams of youngsters. Jack Foundation has joined hands with Symbiosis School of Biological Sciences. This is an endeavor for the development, implementation, and advancement for different areas of biotechnology. The aim of the association is to promote, advance, and disseminate the interest of scientifically oriented biotechnology research and application for the benefit of individuals and society concerned. The National Summit will help provide the platform for the quality exchange, research in different areas of biotechnology disciplines. You know, the toll of uncertain outbreak of COVID-19 has affected so many people around the world. Using our football, it would not be better to regulate today's summit and this pandemic, where all are waiting for the COVID-19 vaccine. This is again an application of biotechnology. We are lucky enough to have internet services as a luxury resource to propagate our ideas, regulate awareness programs, share opinions, and help one another, and even after distant award. We would like to encourage all the delegates to participate with the full energy and commitment, and wish everyone a successful, safe, and fruitful summit. And I am very much lucky that we have among us Dr. W. Silvamurthy, who is a well-known scientist and currently is working in Amity University as a president of Amity Science Technology Innovation Foundation as well as DG for Amity Directorate for Science and Innovation. He is a Chancellor for MIT University, Chhattisgarh, as well as he is a distinguished scientist who served as a Chief Controller R&D in particular Life Science and International Cooperation at DRDO Government of India. Sir has accolades with many awards and many government responsibilities, including the Ayush, and he is currently involved in taking the innovations and science to next level. And also, he is propagating his ideas and viewpoints related to Indian system of medicine and also research in biotechnology so that the vaccines and other development is taking place that can be reached to the society at the earliest so that they can get benefited. And audience, just I am as lucky as you are that we are among us, Dr. W. Silvapurti. I, on behalf of the organizing team, would like to invite Sir to give his keynote address and remarks. So, Dr. W. Silvamurti, invite you for your address. Thank you.
Thank you, Dr. Rajput. Very good morning to each and every one of the participants who have gathered today. I see very distinguished resource speakers here. Thank you, Professor Rajput. He has been our colleague at Amity University, heading the department, Amity Institute of Indian Systems of Medicine. And he himself is a pharmacologist and pharmacist. He is a well-known, globally well-known pharmacist. And thank you very much for very kind and generous words of introduction. And I see Dr. Subramaniam, Dr. Chakravarti, and many other very distinguished resource speakers who are going to share their thoughts and also their experience, expertise in this field of biotechnology. So I have great pleasure in welcoming very young students, researchers who have also joined us today. The whole purpose of this summit, National Summit, is really to ignite the passion in the youngsters who are now driving the future of biotechnology research and development, not only in the country, globally. So that is why I'm so happy to participate in this National Summit on Innovation in Biotechnology era. And I would also like to place on record my deep appreciation to Jack's Foundation for this initiative. And this is a very important mission you have taken. And I'm sure this will yield very fruitful results in the years to come. Because we are the youngest the country in the world today. So all the youngsters who are here going to be the professionals in biotechnology and related domains who are going to lead the research and development at the global level. So there's my appreciation to Jack's Foundation for this wonderful initiative. And this is being organized in a very opportune time today. Why it is very opportune time? Because the whole country is vibrant. This COVID-19, while it has brought a large number of challenges, mortality, mobility, impact on economy, impact on the social well-being, social infrastructure. So everything has been disturbed. But then it also gave us an opportunity for looking at, for introspection, and also looking ahead, how we should move, where we should move, where we have faulted, what we need to correct in our life systems, what are the new normals we should look at, like the one which you are organizing today, how we use the digital platform to connect people across the globe. So you can just be there anytime virtually through this digital platform and listen to wonderful lectures being organized. So that is why this COVID-19 has also given us great opportunity. And this is yet another opportunity to look at what is the trajectory in which the country should move in the field of biotechnology. And also the country is very vibrant with the government giving a focus on research and development, the Atmanirbhar Bharat, looking for self-reliance. Honorable Prime Minister is keen to promote the young startups, Stand Up India, Startup India, and also Make in India. All these are not only slogans, but actually these are the driving forces, prime movers, which will actually give a new energy, enthusiasm, vibrance for research and development in the field of biotechnology. So this summit is taking place at a great opportune time. The other opportunity window is that the 21st century belongs to life sciences. 19th century physical sciences, the 20th century belongs to engineering sciences, but 21st century belongs to you. There is life sciences. A lot of exciting things are going to happen in the field of life sciences, particularly in the field of biotechnology. But here, life sciences combining with technology is biotechnology. Biotechnology. So that is why this is going to be that is why you have named this as biotechnology era. We had agricultural era, 15th, 16th century agricultural era, where we were looking at how do we cultivate food. Then came industrial era between 1870 to 1960, where a lot of the industrial growth occurred, and so steam engines, connectivity, so many technologies came at that time for industry. So industrial growth, industrial era occurred actually from between 1870 to 1960, where India did not play a very important role. But information era, which came between 1960 to 2000, India played a significant role. Maybe that we didn't develop a high-end software or hardware, but we applied the information technology in service sector to produce the wealth for the country. So 60% of GDP actually came from application of information technology into the service sector. So we feel that 60% GDP and GDP growth rate, it provided a lot of impetus, information technology revolution, information era. Now we are up from 2000 onwards, the 21st century, we are now in the life sciences era, biotechnology era. That is why this conference is very important. This summit is very important. And we should look at where do we focus, what is the emerging needs. And this COVID-19 has also brought in, we need diagnostics, therapeutics. So that is where the biotechnology comes in a big way. So 21st century belongs to life sciences, and in which biotechnology is going to play a pivotal role. In this era, India is going to play a leadership role. Why I'm telling is that India is going to play a leadership role. We didn't take part in industrial revolution. We did use the IT technologies in information era for wealth generation. But in the new knowledge era, where biotechnology is going to play an important role, in which India is going to take a lead role. That is where this summit is important. You are important, we are important to see that how do we synergize all our efforts in a concerted, orchestrated teamwork as an Indian team to produce new innovation in biotechnology. What is the advantage India has today? Not only the young population who is creative, innovative, minds who are trained in biotechnology, who are getting trained in biotechnology, very important resource. Because the most of the innovative, creative phase of one's life is actually between 18 to 35. After that, you build on your experience. So the most creative and innovative phase of one's life is between 18 to 35. That population is here in India. So that is advantage India number one. The second one, we now have trained the human resource at senior level, like I see Subramaniam, I see Chakrabarti, I see many very Rajput, I see many distinguished people. So we have trained the senior people to mentor, guide, facilitate, catalyze. So that is another advantage India, that you have already well-trained people who has postdoc experience across the globe, and here they are in India. Then the other opportunity is we have created the infrastructure. If you see, go to any biotechnology lab, it's very well equipped. There's a BSL2, BSL3 we have, BSL4 we have. So you name any facility you require, whether it is fax, the flow cytometer or confocal microscope or fermenter, you name anything what you require for biotechnology infrastructure. Today it is available in the country in a horizontal growth has occurred. So we have the human resource and we have the infrastructure. Then we have also got a government of India has established a separate department, Department of Biotechnology, which is actually to give a focus, directions, policies to enable the emergence of new technologies. So you have DBT, and they have many important programs where to link the industry with academia and also to generate new ideas into blossoming into a technology. So all this DBT is facilitating that to advantage India. The other one is very important is you need industries to translate, productize, upscale, manufacture and commercialize. Today India is blessed to have great many industries in biotechnology. Like, for example, Bharat Biotech, Shanta Biotech, Biocon, India. Name, there are a number of them. I don't have to name everything. I just mentioned a few. And there are many biotechnology industries. And if you look at the startups, many startups are coming in the field of biotechnology domain. So you have a vibrant industrial ecosystem in the field of biotechnology. Advantage India. Then the new policy, science, technology, and innovation policy, which the central government has promulgated and is now being discussed across the nation. Now that policy will also enable the industry, academia to blossom, to bring innovative technologies, disruptive technologies. So that is why this is advantage India. So this summit is actually taking place where India has such a great advantage, which should be harnessed to become leader in biotechnology, not just follow up. In certain niche areas, India should be known first time for in the world. So let us dream that how do we achieve that? So this summit should actually think in that direction, that how do we become leader in biotechnology, having all this advantageous position in the globe. And if you look at the market today, biotechnology market, it's multi-billion dollar business. And then India should take the major share. And we have biodiversity. We should look at herbal medicine. We have a hotspot in Northeast, another one in Western Ghats. Biodiversity. Harness it for biotechnology innovations. So the global market is huge, and Indian share should be the huge one, maybe the best, the largest one in the biotechnology market. We should develop that. And growth rate is also projected growth rate is very high. So we need vaccines, we need generics, we need herbals, so nutraceuticals. Tremendous opportunity, youngsters. I see the young, brilliant researchers. You have a huge potential. Now go full steam, go full steam with the thrust. Then if you look at when I was in DRDO, I spent 40 years in Defense Research and Development Organization, pushing the life sciences for the health of the soldiers who have to operate in high altitude, cold, desert, underwater, aerospace, low intensity conflict. So I used biotechnology to develop their food. For example, in high altitude agriculture, I used biotechnology to produce 5,000 metric tons of vegetables in sub-zero temperatures. Then we used hybrid cows to develop milk in high altitude. So, such kind of innovation. Then one important thing which we did there in DRDO was biotoilets. How do you handle the human waste in high altitude, in cold temperature, sub-zero, -20, -30? Human waste will not be biodegraded. So we went to Antarctica, brought a consortium of psychrophilic bacteria, cultured them in the laboratory, and provided the right kind of the ecosystem there, the temperature, the pH, optimized those conditions in a bioreactor, and ultimately we developed an inoculum, which we put it in toilets, so it became a biotoilet. You see in trains, what you see in biotoilet is DRDO developed, and I coordinated that research in DRDO. And today, 11,500 toilets have been put in Lakshadweep Islands, and 50,000 toilets have been put in the trains, which has been retrofitted with DRDO developed biotoilet. and we have put for Javans in high altitude, it's working at sub-zero. So this is biotechnology. This is biotechnology. How we can handle a situation where there is no solution, but we find the solution through biotechnology itself. And similarly, now when I look at the Amity, I have now joined Amity during the past about 8 years. I'm with Amity University, driving research, innovation. We have 11 universities in India, 17 campuses abroad, 28 schools, 1,75,000 students, 10,000 faculties, and 5,000 PhD scholars. Huge Amity universe. So I look after all campuses for research and innovation as the president. And also we have a campus in Raipur, that is Chhattisgarh. I am the chancellor for that. So why I'm telling is, in all places we have created biotechnology. In all campuses, 11 universities here and abroad. So biotechnology is there in all our campuses. Knowing the importance of this, we have created. And currently in Noida campus alone, 2,000 students in Biotechnology Institute alone, 2,000 students, 135 faculties in one department in one university we have. So that is the strength we have created in terms of human resource. So we will be churning out every year 10,000 biotechnology students in undergraduate, postgraduate, and doctoral level. Then we have created the research infrastructure, high-end infrastructure for biotechnology. You should come and see. I invite all of you, build partnership with us, and we will be opening all this to, it is open to all of you, so please join us. And when I look at the R&D contributions in biotechnology domains, because we have many other domains, I will for the talk today, I will look at many innovations. I'll pick up a few to illustrate how biotechnology research is happening in Amity universities. The first one is, see, there is a lot of change. You have to do cropping at even in difficult places where abiotic stress is there, like high temperature, low temperature, salinity of the soil, alkalinity of the soil. So which is all challenge to the crops. So how do we give the tolerance to abiotic stress? So we looked at microbial technology. We cultured the Piriforma spora indica, which is P. indica, which we have named as root tonics, but actually it is Piriforma spora indica, which we cultured initially in the shaker, beaker, initially in the small fermenters, then scaled up. Today it has gone to 5 industries. Industries have taken, we have optimized the condition for production in the bioreactor. So even including 2 companies abroad. One is Taiwan-based company, Sinon Corporation. The other one is Canadian company based in Calgary. So we have these technologies and 5 industries in India. So look at that. So this will give protection against the abiotic stress. And this is a root endophyte, which we have through biotechnology research, we have now given to the farmers. The other one is purity of inoculum. In the industrial biotechnology, when you are having a vaccine production or when you are having the microbes put there, how would you know the purity of your inoculum, which is there? How do you identify the purity? So we have developed a method in the Amity Institute of Microbial Technology. Then we have a lot of focus on herbal medicine for various applications, like, for example, aspergillus, then also for H. pylori. And we are looking at herbal medicine in a big way. And we have also combined herbal medicine and the allopathic medicine together in our biotechnology research. For cancer, now Amity University is focusing on cancer stem cell, because cancer is recurring. Why? Because you may do surgical removal, give chemotherapy, radiotherapy, but you may not be able to kill all the stem cell for cancer stem cell. So we are now attacking cancer stem cell instead of cancer cell alone. So this will prevent the recurrence. So how are we doing? So we are doing a known anti-tumor agent, curcumin and folate as a triple conjugate we are developing. And this triple conjugate, what is the advantage? So we did in silico modeling first, then we went into synthetic chemistry. To make the conjugate is a big challenge. You have a synthetic chemical, you have a herbal curcumin, and then you have folate. So we first developed in silico modeling, then synthetic chemistry to develop the conjugate. Making conjugate is a big challenge. Then we did in vitro. Now in vivo studies have shown outstanding results. So you will prevent the recurrence of cancer cell. So we are attacking cancer cell through biotechnology research. So what I mean to say is you should integrate the traditional system, traditional knowledge, curcumin, and a known anti-tumor agent. This is how new things, novel things, disruptive things will come. So that is what happens in MIT University research. Then we have also developed a artificial intelligence based platform using bioinformatics. Bioinformatics is very important. So what we have done, we have developed a AI platform, artificial intelligence platform, but in which you can identify if you have so many molecules, which is the candidate vaccine, which is most likely to succeed. You can narrow down by doing a in silico biology and looking at the bioinformatics. We have developed an AI platform. So all the pharmaceutical company which are in the development of vaccines, including the COVID-19 vaccine, and Baylor College of USA in Texas, we are now partnering with them in the AI-based platform for candidate vaccine, which will also give the potential efficacy and toxicity. So you can predict them by using this AI platform. We have a strong nutraceutical research group in MIT University. And so there are a lot of exciting things happening now in MIT. Now, just to conclude with another couple of minutes, what are the emerging areas in biotechnology? What are the emerging domains? Gene editing, the Nobel Prize, it went for the gene editing, CRISPR-Cas. So this we are already using in MIT University in very many research, and this is going to be a potential area. So I would like to tell youngsters, gene editing, gene therapy, and GMO will assume a great focus in this biotechnology era. Synthetic biology, computational biology, again will assume a great relevance. Synthetic biology is going to be the one which has got a double-edged weapon. You can also give therapeutic remedies. You can also use it as a biowarfare agent. So we should not use the second one. We should look at synthetic biology as a tremendous potential in biotechnology. Then we must also look at how do we use AI platform for diagnosis. Like we are simple screening. We are doing an experiment. We are doing a project on with the hospitals that cancer cervix. How do you screen by just in the field, not bringing them and then taking a pap smear and then looking at this? We have developed a simple algorithm which can detect the normal cell, the reference, and the cancer cell in the early stages, different stages of severity. So we have developed that platform. So AI platform to early screening, large scale screening, even in villages, using this visual, the system, optical system, we have developed that. So this is another important AI application in the biotechnology is going to be increasing tremendously. So that's another opportunity. The other one is biofuels. Certainly biofuel is going, that third generation biofuel is going to assume a great relevance. So this is another biotechnology exciting area one should look at. Then convergence technologies will be a focus. The bio will not stand alone. It will be linked with nano. It will be linked with info, IT. So it will be nano, bio, info convergence. So now start preparing that in the biotechnology, how do we integrate with these two disciplines? So convergence technologies will be the focus. Then in cancer, the immunotherapy and the cell therapy will assume great importance. Now it is gaining momentum. It needs a lot of improvement. Biotechnology has to make a big contribution in this area. So these are some exciting things which will happen. So my compliments to all of you for this initiative. We have to do. We should not only keep talking. We have to do it. We have to keep, we are good planners. We create ideas, but somebody else do it. But now we should change that mindset. We also give ideas. We also do it and bring the product. So ultimately, all of you sitting here should aim, what is my ultimate deliverable, which is going to come through my research, should be visible in your mind. Then take actions, a strategy, plan your strategy to reach that goal of a deliverable coming out of it. So this is how we must work. Our time of only publication, yes, publication is important for our promotions, growth, invitation for conference, fellowship in academies. But what is the return on investment which we give to the government, to our country, is through deliverable. So I would request you, all these people, integrate yourself, do a parallel processing instead of sequential processing. Unite as a big team, in silico modeling, synthetic chemistry, then in vitro group, in vivo group, clinical trial group, formulation group, integrate together as a group, big team. Then you can bring the product much faster. So this should be the goal, and I am sure this forum will give such kind of strategies. I will be very keenly looking forward to the outcome of this, and my best wishes to all the participants, and particularly all the very senior people who have joined us now. I can see Dr. Praveen Pordar and Jyotimoy Aish, who is going to speak as well. So all of you are very important, but let us not just only keep talking, delivering lectures, very important to disseminate, but let us actually make plans and strategies for the country, for bringing a lot of exciting products, technologies, processes, bioprocesses, which will generate wealth for the country, which will solve problems of our country. So best wishes for the successful outcome of this conference. Thanks to the Jacks Foundation for the great opportunity you have given me to share some of my thoughts and little experiences during my almost now it is 70 years, and of which 40 years experience at DRDO, now 8 years in Amity University. And thank you very much for this opportunity. Then one more message I want to convey to all of you, just last one, only 2 minutes. So, Honorable Prime Minister has now promulgated a concept of scientific social responsibility. And you may be aware that they have appointed me as the chairman to make the national policy on the scientific social responsibility. All of us have a social responsibility. This we must remember, not only in our profession, we should go out beyond the call of the duty as a scientist to look at the societal need and go beyond the call of your duty. And this policy is already under consideration. It's going to the cabinet. The cabinet note is being circulated for interministerial consultations. We have already done a national consultation. Now this will be first time in the world a national policy will be promulgated on scientific social responsibility. So your country, our prime minister has taken this initiative to start scientific social responsibility. So when you are deliberating, keep that in mind, the social responsibility of all of us. So best wishes and thank you very much.
Thank you very much, sir,
Thank you, Doctor.
for participating and giving us blessings as a chief guest. And your words of wisdom always inspires, motivates, guides, and ignites the young minds. And I believe that with your blessings, the youngsters, the participants, the speakers will brainstorm and bring some out something which will definitely will be the life changing. And as you are heading for the very important mission of Prime Minister, that social responsibility stands for social responsibility. That is the amazing task. And with your energy and dynamism, I believe, and we are your soldiers, and definitely will continue wherever is required. So thank you very much for the servers for blessing us and igniting us. So now I will request Ria to take forward this important evening. Thank you.
Thank you. Thank you very much. God bless.
Thank you, sir, for your gracing us with your valuable presence here. Now, I would like to introduce you to our chief guest, Sri Satyendra Kumar Jain, Honorable Cabinet Minister of Delhi, Government of India. Our ministers, Dr. Harshvardhan, Satyendra Jain, are unable to join today due to some prior commitments in Parliament and some health issues. He has sent his warm wishes and blessings to the summit. We also have warm greetings from Sri Raghuraj Singh, State Minister Labour and Employment, Uttar Pradesh, for this national summit. I request Ms. Sania Bhagat to please play the video.
विद्यार्थियों की ऑर्गेनाइजेशन जैक्स फाउंडेशन द्वारा मुझे निमंत्रित किया गया, इसलिए सर्वप्रथम सभी को बधाई, साधुवाद, धन्यवाद कि आप लोगों ने मुझे निमंत्रित किया, इस कार्यक्रम के लिए मुख्य अतिथि बनाया। मेरा आप सभी से आग्रह है कि इस कार्यक्रम को दिन दूना रात चौगुना तरक्की के आयाम के माध्यम से विश्व लेवल की ये संस्था बने। जय हिंद, जय भारत। विद्यार्थियों की ऑर्गेनाइजेशन
I heartily thank all our chief guests and ministers for their blessings, even their absence. Now, I would like to invite Dr. Vinay Kumar Rale, Director of Symbiosis School of Biological Sciences. Dr. Vinay Kumar Rale is MSc PhD Microbiology from University of Pune. He has completed his first postdoctoral from Germany. With versatile and rich experience spanning over 40 years in teaching, research, administration, market, and techno-entrepreneurship, and an excellent practice with primary teaching and research institutes with diverse industries. Dr. Vinay Rale is back again in a new challenging role as Director of nascent Symbiosis School of Biological Sciences.
Yeah, thank you, Ria. Am I audible?
Yes, sir.
Yes. Thank you, Ria, for the introduction, kind introduction. I would like to also thank Dr. Selvamurthy for raising the curtain and giving a beautiful bird's eye view of the development of biotechnology across the country, with special emphasis on the Amity University. So I am very familiar with the Amity University activities, but it was a good thing to listen to him after a long, long time. And of course, Dr. Rajput for giving a preamble. May I start sharing with my slides? I'll start sharing with my slides. Am I still audible?
Yes, sir.
Yeah. Yes. PPT for audience. Yeah, is this visible now?
Yeah.
Yes, sir.
I've entitled my talk, which is very brief. I have entitled my talk as Imagination and Necessity, which is the linchpin of innovations. And I would like to not only restrict to the imagination and necessity for innovations, but even take the wider range from serendipity to even the pressure of doing something, pressure of finding out a new development. And that is also included in that. So you have at the one end serendipity, and the other end you have a pressure. I'll go to the next one. This is me. This is me. I'm a microbiologist with about 45 years of experience in teaching, research, consultancy, and industry. I do own an industry and a good deal of experience abroad in the leading University of Göttingen in Germany, and then a good deal of stint at University of Pune as professor and head. I'm back again with this Symbiosis. I'm sure Symbiosis International University is a very well-known entity in the Indian scenario. Established in 1971, it is spread over the entire country, with plans to even extend to overseas campuses. But I must confess that we are restricted as far as science is concerned, hardcore science, biotechnology is concerned. We are restricting to only the Pune campus, the Pune Lavle campus, which is a new campus. Why Pune? Because Pune is a hub of leading national institutes and industries. So for time being, we are restricting ourselves of staying in Pune. And maybe in the future course of time, we will extend our biotechnology and other relevant areas into the other campuses. So I go to the first slide now, which is a preamble. The commercialization of scientific and technological knowledge, ideas, and competencies, which are emanating from institutions of higher education like universities and research institutions, is a much talked about discussion or much talked about topic in the recent past. And there is a good deal of recent transformation that has taken place in this direction. In the Indian context, there is a usual style of referring as innovations, innovations, innovations. There's also a thrust for transformation, which has originated from market-oriented ideas and practice. That is what I was referring to when I said serendipity to the pressure of innovating something, pressure of developing a certain equipment or a certain technology. Now, at the same time, we should not forget the question that to what extent are we then as compared to the past benchmark? Because we were old style British universities some time ago, and there is a great deal of transformation in the Indian university scenario now. We are all competing with the leading universities in the world, and even Symbiosis is attracting international students. We have currently 85 countries represented in Pune campus alone. So a good deal of inflow from the other developing and developed countries is happening. So in that question, that context, how are we different from the past benchmark? This is a question we must ask ourselves. Now, the innovations is something different in the US universities and Europe. Say US universities, mid-19th century, and in Europe, more distant, even older than that. And I will substantiate this by giving specific examples in the next few slides. In the recent past, again, in the context of research, there's a concept of triple helix, which is the commercialization in the third mission, complementing traditional tasks of the university system, which are only restricted to education and research. So we have a third mission in this added, and that is called as the commercialization. The universities are now taking a good deal of leap into commercialization, and which is a very welcome step in the Indian scenario. In simple words, university is no longer a mere secondary actor in the process of innovation and economics development. So university is something now comparable to some of the leading research centers across the country and even the government organizations. So university now is a structural element along with the government and industry. I'll take this idea further. In the next slide, the triple helix configuration is explained here. This is a loose configuration. Nothing much happens here. That's why it's called as a negative sort of a thing. But then you have a very intense positive interaction happening between university, industry, and the government. And this is an area which will probably lead to commercialization, lead to innovations. The business model of the mediated between technical and economic domains is represented here. You have the technical inputs, feasibility, performance being assessed at the university level or research institution level. It goes through a business model where it looks at the market, value proposition, the value chain, cost and profit, value network, competitive strategy. And eventually, it transforms the information, the data collected into economic output, which is adding value, adding price, and naturally giving profit to the organization, to wherever it is being transferred. This is more elaborated, for example, in the triple helix, which is happening at all levels here: novelty production in science and technology, wealth generation in industry, you have legislative control through the government, you have a knowledge infrastructure, you have innovation happening here, and you have the political economy. But the resultant of all this is the patents, which is a very important aspect in the university education. And of late, the Indian universities have been doing a great job competing with the leading universities in the world as far as patents are concerned. I'll give you a few examples of this in my later talks. So eventually, the idea is to have a entrepreneurial university. These are the factors of creation and development of entrepreneurial universities. And these are the formal factors, these are the informal factors. And besides the, the traditional areas of teaching and research, this is an entrepreneurship being developed or promoted by the universities by having incubators or by having universities being participants as equity participants in the commercialization of the processes developed in their premises. We have developed such processes here in the symbiosis, and some of them are now on the verge of getting into commercialization. So I'm very happy to mention that. They are specifically in the nanoscience, nanotechnology, in the field of vaccines, particularly the veterinary vaccines. These are the areas that we're focusing. We're also focusing on the bioprocess engineering at the same time. I'll give examples related to my first slide where I said that innovation is nothing different. The idea, the word innovation has caught up the imagination of all people, but there's nothing new in that. Innovations have been happening all across the country, especially in Europe. In the 18th, 19th century, you have the well-known case of Emil von Behring. Everybody knows who Behring is, and this has more relevance now in connection with the COVID-19 vaccine development, where you could probably have the serum or the plasma being incorporated and then develop an immunity. So the idea of this vaccination in this particular format, after having learned at the feet of Robert Koch, Behring developed his own company. So you have now an example of the university education and research transforming into the industry. So you have now a Behring Verke, which is very leading organization in vaccine production. This is how Behring appears. And you have another example from Germany, von Siemens, Werner von Siemens. And I'm sure you're all conversant with the developments Siemens have done, so much so that there is a Siemensstadt now in Berlin, named after the company. But then Siemens at the same time around 40s, if you're not aware, had a great deal of role in developing an electron microscope as a competitor to the Philips company. So Siemens and Philips were both competitors in developing the earlier TM models, which came into the market. Another example, of course, is known is the X-rays. Again, Wilhelm Conrad Röntgen was a professor in Würzburg, developed X-ray technology, and now we all know that X-rays is a very essential thing in the diagnostics. In fact, in Germany, people don't call it as an X-ray, they call it as Röntgen. So how a name becomes a word or a noun is a very well-known example in the form of Röntgen. Why don't you do a Röntgen? Then why don't you do an X-ray? That is what they mean to say. That's in German style. So you have a Röntgen museum also in this area. I'll give one more example, like the antibiotics development that took place in England, and then Florey & Chain took it to the NRRL in US. At the same time, the development took place in Germany also. So you have Hans Knoll transferring himself from Berlin to Jena, and then interacting with people like Carl Zeiss and Otto Schott. I'm sure you know who Carl Zeiss is, and I'm sure you know who Otto Schott is. Otto Schott developed a glassware company, and Carl Zeiss developed a microscope company in collaboration with Ernst Abbe. So here you have Carl Zeiss and Ernst Abbe at the University of Jena, collaborating with the industrialist Carl Zeiss and developing a world-renowned company in the microscopes. And around 1945, and after the World War, Carl Zeiss was split into two. The Allies took part of that into West Germany, and the part remained in East Germany. But in 1990 unification, both the companies came together again with a stronger force. So Zeiss microscope is something like a prized equipment in our laboratories. So here's a well-known example of Carl Zeiss and Ernst Lavie. Innovations happening. Otto Hahn, again, the generator of the atomic energy, so much so. But then again, the initial workplace was the University of Göttingen, which is a hub of more than 65 Nobel laureates. And I was very proudly associated to the Institute of Microbiology in Göttingen, which has many renowned scientists its credit. So I will take you to another example of innovations, how balance is developed. Sartorius in Göttingen, with which this is a company I interacted with for my postdoc work. Sartorius was a mechanic in the University of Göttingen, developed this simple balance, and eventually turned into a good, world-renowned, multi-billion company. So here is the Sartorius original company, which is still there as a head office. I interacted with this building in a new format for my work on acetone butanol fermentations. Just to give you a glimpse of what we've been doing here in Symbiosis, School of Biological Sciences. This is one patent. We recently generated a method of inducing sporulation in Bacillus coagulans. Bacillus coagulans is a very well-known probiotic strain of greater demand in pharmaceutical industry, but they desire this particular strain in a tadpole fashion. And to generate this organism in a tadpole fashion, which is a sporulating form of Bacillus coagulans, it is very difficult. So we developed a medium, and then this now is being patented. This is already published. It's in the process of being patented in the Indian scenario as well as in US, Japan, and China. There's another patent that has also come up from our department. That's the method of isolation and enumeration of yeast from diverse milieus using a suitable medium, that is the modified molybdate agar. And many times we come across difficulties in selectively isolating yeast from different environments. So we have overcome this difficulty by developing a modified molybdate agar. It is also getting patented. It's already published. Now, in the last two slides, would also like to focus on the future focus. But at the same time, I must happily mention that from our department, two new genera have been recently reported. They are Corynebacterium godavarianum and Chakravartiya godavarianum. Chakravartiya, we gave the name of the famous microbiologist working in Chandigarh. His name is Tapan Chakravarti, now currently stationed in Pune. And we named this genus after him in order to honor him. And many of our interactions now are happening with the Symbiosis Hospital now, which is at Lovely. It's a state-of-the-art hospital, and a medical school, which has just commenced its operation. This is the third medical school in the country, which is only meant for women. So it's called as Symbiosis Medical College for Women. This slide shows biotechnology advances are rapid. However, return on investments is low, and innovations need to be protected as IPR helping to generate ROI. This aspect is going to gain a good deal of prominence, and as Dr. Selvamurthy suggested, that IPR is a very important aspect. When something comes out, it needs to be transformed into IPR to protect the scientist, to protect the knowledge, and also to generate money, finance out of the development. The second aspect, which is going to be a future focus, is critical analysis of biotech products to determine and demonstrate real and measurable value. Critical analysis is obviously required for seeing to it that they add value in the commercial market. There is a yawning gap always between research in labs and the actual market. I have a personal experience of that, having produced fruit yogurts and bread spreads, dairy products in India. They were all import substitutes in the country then. And then I learned how research has to get into the field and compete with the well-known companies. The future R&D will be also networked, that is involving academics and others in depth understanding and research for. So it's going to be a collaborative work. Nobody can work in silos or in isolation. People have to come together, and that's the reason why we are primarily focusing on staying in Pune, so that we have a good deal of opportunities of interacting with the leading organizations in Pune. The third important aspect for the future focus is that the regulatory bodies will intensely focus on safety, efficacy, and quality of life. So for that, massive data analysis will be the basis for establishing these processes. This is going to be of great importance in the years to come, not only in terms of vaccine, but any biotech product that you develop. Again, Selvamurthy has also emphasized that how government is coming forth, government support and eager graduate workforce will form the linchpin of biotech industry. So both together, it's a collaborative attempt, but the government is more than anything else very liberal in the recent past of funding for research. At the same time, they have also become stringent in asking for returns. What's the outcome of the research projects? So as compared to the olden times, government support is very liberal now. So government support and the eager graduate combination is going to make wonders. The future focus that we've been experiencing in the recent past is the major startups, more than 50% are in the diagnostics, drugs and medical devices. Agricultural biotechnology is about 14%, bioindustry is 3%, bioinformatics is 1%, and services is 18%. So these are some of the major thrust areas, but we have a good deal of chance, Selvamurthy says, bioinformatics, giving a thrust on this. Even services, university can offer services to the industry, and that we've been offering now, sitting in Pune in Lavade campus. Then the easy accessibility of funding from government industry. This is again related to the earlier part that I mentioned, government support. So this is also happening in the recent past. And of course, the last but not the least is the consumer's demand, which is linked to innovations. Ideas must generate and that get implemented. It is not leaving haphazard, half-baked products. They must get into the market. They must see the light of the day. So consumer demand, that's a pressure. That's a pressure of innovations. And that is how the universities and research institutions, particularly universities, have to meet these demands and then reorient them in order to meet, in order to come out with innovations. With these words, I would wish to thank the JAX Foundation for giving an opportunity to Symbiosis International University for being a collaborative partner in this two-half-day workshop or summit. And I'm sure with the stalwarts that will follow me and tomorrow, today in the afternoon and tomorrow, they will contribute very heavily and significantly. And the students, particularly the postgraduates who are on the verge of selecting a career in future, will be very happy to listen and reorient themselves in order to find out which areas they are comfortable with. So with these words, I would wish to again thank the JAX Foundation, everybody else here, and sign off with happy listening to all of you. Thank you so much.
Thank you so much, sir. I would like to express my gratitude for portraying such an enlightening message. Now, we would like to start with today's plenary sessions. I would like to introduce today's moderator of the session, Mr. Aditya Acharya. He is an executive member of JAX Foundation. Currently, he is a member of Global Microbial Identifier Group, Singapore, active member of Federation of Asian Biotechnology Association and Indian Microbiology Society. He is currently doing research in the field of computational drug designing to find the lead candidate for SARS-CoV-2 virus. Now, I would like to call Mr. Aditya Acharya to take forward the event. Over to you, sir.
It's my honor to be invited here, first of all. We are living in an era of new ideas and creativity and for innovation which is the practice of these ideas in shaping a new product, process, or both, where biotechnology is immensely related with our lives. Standing today in this national summit, the theme for which is innovation in biotechnology era, the need of the hour. So let's start with the plenary sessions. I would like to call our first speaker of this beautiful morning, Dr. Jyotirmoyi H. Ma'am, Professor at D.Y. Patil College.
Yeah. A very good morning to one and all. I hope I'm audible.
Yes, ma'am, you're audible.
Yes. Thank you, Aditya. I would like to thank Jack's Foundation for giving me this opportunity to speak something about my work, definitely. Now, I know that we all have been facing this pandemic issue since past 7 to 8 months, actually. And I think all of you must be agreeing that, we have been listening to a lot of webinars, seminars, and everything. But what was lacking is the student participation, actually. Now, student participation, basically, now that is the thing that I'm looking forward in this meet, because this is the summit for you people, for you students, actually, right? So again, I'm looking forward for the poster presentations, for the group discussions, for the brainstorming session, and definitely for looking to all the sessions by senior scientists, by different faculties and everything. I would like to present my work. Okay, I will be sharing my slide. Aditya, just 2 minutes. I'm sharing my slide.
Yes, ma'am, no problem.
Yes. Just give me 2 minutes. There's some. So is my slide?
No, ma'am.
No,
There's
it is not visible.
You have to present the screen, ma'am. Present
Yes,
now.
yes. I tried that. I just tried that. Just give me 2 minutes.
Yeah.
Yeah, it is opening.
Yes, screen is visible now.
Yeah, screen is visible.
Yes.
Okay. Yes, Aditya. Okay. Very good morning again. So I'm Dr. Jyotirmoyi. I'm working as an assistant professor in the School of Biotechnology and Bioinformatics, D.Y. Patil Deemed to University. I'm a postdoctorate. I have done my postdoc from ACTREC, and I'm a PhD from IGIB Delhi. So the topic that I will be talking about something is all about the drug repurposing in oncology. I think Selvamurthy sir and Dr. Vinay Kumar Rale, they have really given a global scenario of whatever the opportunities and everything are presenting regarding biotechnology. Okay, so something that I would be liking to discuss about my work in the School of Biotechnology and Bioinformatics, that is going to give the students an overview that what kind of a work we do in our system. Now, repurposing. The word repurposing, what do you exactly mean by the repurposing? As the word itself is going to mean that I'm going to use something I'm going to use for something different purpose, right? So it is the use of something for a purpose other than its original intended use. So before going to the scientific applications and everything, let's look down at some of the repurposing things that, that is famous. Okay, now if my slide is visible, now can you see there's an old there is an old ladder that have been changed into a bookshelf, right?
Yes. It is still on the file manager only. It has not changed to your presentation yet. We are able to see only the set of files in your laptop, not the PPT.
Okay. The slide is not visible?
No, the slide is not visible.
Okay, I will again try. Sorry for the technical.
No problem.
I will just stop presenting. I will again try. Sorry for the technical. Is it fine?
1 second.
Aditya?
Yeah, yes.
That is visible. All the slides. Okay, fine. So sorry for the technical glitches, actually. Okay, so I was talking about the repurposing things that I'm working on this field of repurposing, that is a drug repurposing. Now, these are the some of the examples of the repurposing thing that I have just taken, so that it is going to give an idea that what kind of a repurposing can be done. Now, can you see a ladder is there where there's an old ladder where the books are being arranged, or there is a tennis racket where the mirrors are being made, right? And there are some the old bulbs are there which has been turned into a lighting lamps. Coming down to, there's a piano in which old piano, it has been turned into a bookshelf. Now, can you imagine the middle pictures? This is something, these are the disposable spoons that we used to long time use in our parties and everything, and a beautiful lamp has been made out of it. And out of that, and again in the right one, there's an old TV where the aquarium has been made. Now, what is my intention to show you all this actually? Because there is something, there's an old thing. There was an old use, but it has been used for a new purpose kind of a thing. So coming on to my topic, that is a drug repurposing. What do you exactly mean by a drug repurposing? Drug repurposing, it would be also called as a drug repositioning. It is also called as a drug reprofiling or a retasking, right? And again, it talks about a strategy that is for identifying some new uses for the approved or the investigational drugs. So that holds the most important part of the drug repurposing, is that already the drugs which are being known for, the humans has been already used for some different disease, it can be used for a different therapeutic purpose. So it involves investigation of the existing drugs for the new therapeutic purpose. Now, talking about cancer, I'm not going to go into the detail because already all the scientists and everybody, we know about it, like what is the difficult part of the cancer. So I'm going to tackle about that particular thing. Now, cancer, we know it's a group of disease. We know that there are so many different types of cancer and everything. The major treatment modalities which are present, it includes basically, depending upon whether it is a benign or a malignant, it has metastasized or what is the situation, the chemotherapy or the radiotherapy or the surgery is being prescribed, right? And some of the best anti-cancer agents which are used for the breast cancer, lung cancer, cervical cancer, and the list is endless. The examples being your cisplatin, tamoxifen, erlotinib, gefitinib, and there are so more. What is the problem that the clinicians are facing? I would like to point out that particularly because that forms the basis of my research that we do in our DY Patil University. Okay, the phenomenon comes out that is a chemo resistance in the cancer. I think most of the senior scientists who are working about it, also Selva sir just talked about, like, they have been targeting the cancer stem cells. Yes, sir, that is one of the most important area. And again, one of my team is again working on that particular part as well. It was very nice to listen to you about all these things. So chemo resistance is the ability of the cancer cells to evade or to cope up with the presence of therapeutics. That means the cancer cells have become really smart. With the prolonged use of all the chemotherapeutics, these anti-cancer drugs, they have developed this chemo resistance, and as a result of which it is not showing that good promising results. Chemo resistance, it also causes some disease relapse and metastasis, and the challenges that thereby it challenges the improvement of the clinical outcome of the cancer patient. So what is the need of the hour currently is to find out some novel therapeutic approaches for the cancer therapy, because we know because these days I think we get to know that there are a lot of cancer cases and everything. We also get to know, okay, now the chemotherapy has started, but the side effects are so huge. So what can we do so that to reduce that side effects of these chemotherapeutic drugs? So let's talk about something. That is the reason I have initiated my work on this drug repurposing. Now, what is the difference between a traditional drug discovery versus a drug repurposing? Now, whenever you go for a traditional drug discovery, there are so many steps involved. It almost take like 8 to 10 years for a drug to come into the market. Like starting from the basic study, a drug designing, you need to do experiments in the in vitro, followed by in vivo validation, and there are some phase 1, phase 2, phase 3 clinical trials, and then it comes to the market, right? But when you come down to this drug repurposing, okay, now drug repurposing is basically, now already the drug is in the market, already the people have been consuming it for some different purpose, for some different disease. So already the safety and the issue has been covered up. So what left is that you need to reposition, you need to repurpose the drug in a different disease. That is the most important thing, and that is the reason the drug repurposing, the scientists are favored. So scientists favor drug repurposing because we have enormous bioinformatics knowledge. We have enormous genomics data. This bioinformatics, it gives us starting from nowhere, at least we have something so that we can ponder upon that, we can do some analysis, and then we can further go for our in vitro data. So researchers, they need 1 to 2 years to identify the new drug targets and 3 to 4 years to develop a reposition thing. So thereby, again, what we do, so that is the thing that also even I do in our lab, that we take an approved or abandoned from a database of a non-cancer drugs. We do try to do a virtual screening by using the bioinformatics approach, by exploring the drug target disease relationship, right? And thereby, it might lead to the discovery of some novel anti-cancer agents. That would be the most important, and that is the thing that we are targeting in our lab. These are some of the examples of the drug repurposing. I've just taken the newspaper article so that it is going to shed some light definitely that how much the drug repurposing is important in the current field of cancer biology. So there are some leprosy drugs that has been used to treat cancer. Now this work has been done is currently being done in the Central Drug Research Institute, Lucknow. Or again, there is a drug which is the dextromethorphan that is a cure for a dry cough. It can be used to treat the type 2 diabetes. And there is one more drug called as a mifepristone, and it is known to induce a miscarriage, but it can also be used as a remedy for a bipolar disorder. Right now, these are some of the important things. Apart from that, this is the newspaper article that the IC1 has released, because we know that we have been going for this COVID-19 situations and all. So thereby this the drug, the antiviral drug or the skin drug, the arthritis, the leprosy, they all, people have been proposing that it can be used for the COVID-19. But again, this was long time back when there were a lot of research were happening on coronavirus. Now this was the thing, I think most of you all must be knowing that how much the hydroxychloroquine was in the news. Now hydroxychloroquine is an anti-malarial. Okay, so it's a drug that is being treated to cure a malaria, and then people have proposed that okay, now hydroxychloroquine can do wonders for the treatment for SARS-CoV-2. But again, there was some limitation because in vitro studies were there. It reported the antiviral activity, whereas there was not much of in vivo evidence was there. That is the reason it was not recommended to use for the current pandemic outside of the clinical trials. Now, the methodology that we use, we take a library that we already discussed. We take a library of different kinds of drugs. It could be antipsychotic, it could be anti-inflammatory, it could be cardiovascular disease, depending upon, from the databases. Then we do a kind of a drug repurposing, and we try to identify that, okay, out of all this collection, which one can be proved to be a useful or a noble drug. Strategy that we use in the lab. We do an extensive literature database. There is a big database again known for the drug repurposing. We identify the different drugs. These are our candidate drugs. We do a lot of computational prediction tools using molecular docking. There's a combination softwares and everything, and followed by this molecular docking, and then we go for the in vitro studies. So teaching old drugs, new tricks. So these are the different approaches that we do. We do a phenotype-based blinding screening. We do a knowledge database screening. We do also, again, have a target-based approach and a signature-based approach. Because we have so much of data available, bioinformatically, we can approach this data. We can identify, like, okay, now which all drugs could be having a good repurposing potential. So in my lab, few groups are working on a collection of an antipsychotic drug, anti-malarial drug, antiviral drug, anti-inflammatory drug, and we try to see that, okay, from all of this, which one could have a good repurposing potential. Now somebody can ask me that how is it possible? Because cancer is a multi-gene disorder. There's so many, genes that play an important role in the cancer. So thereby, we choose all these genes, we do a kind of a molecular docking, and then we confirm, and then we follow by in vitro and in vivo experiments are also in the plan for the future. Just giving you one example of my data, talking about the anti-malarial drugs. So what we did, we have taken a collection of the anti-malarial drugs, mefloquine, chloroquine, hydroxychloroquine, and everything, right? And then we choose the genes as well. Now I'm just showing you a snapshot of one of the data. I'm not going to, show a lot of scientific data because this is just to encourage you to take up some of the important innovative projects and all. So we choose some of the genes like ER alpha, PR, EGFR. Now these are all important for a breast cancer. We again choose, depending upon our literature search and drug repurposing, we choose 3 drugs. We do a molecular docking. Okay, now these are molecular docking, as most of you will be knowing. This is just to identify the ligand receptor interaction, right? And we do then a comparative studies kind of a thing, like, okay, out of all these drugs, if I have screened for these number of genes, which one is showing me a good, binding energy? So for example, in this case, my data showed like mefloquine showed a very less of binding energy, which means that the association with the target would be very efficient. And thereby, if I'm going to go for my in vitro approach, although it is not very sure, because these are all in silico methods, right? Although it's not very short, definitely everything is going to work properly. But still, it is going to give me an indication that if I'm going to go for my in vitro efficacy, I think it is going to show me some good promising results. So we have chosen this mefloquine, we have taken this MCF7, some other data that like MD, MB2 also we have taken. I'm just showing you some of the data. It's in vitro cancer cell breast cancer model. We used to do a lot of assays like a cytotoxicity assay, a wound healing assay, clonogenic assay, migration assay, as well as a autophagy assay also to confirm. This is just one of the representative picture. For example, if you can see the control cells are there, then the moment I'm going to give these cells, I'm using a cisplatin. So I'm having some collaboration with the Tata Hospital Research ACTREC, and thereby we are working on these anti-cancer agents. So the anti-cancer agent is procured. I have given to the cells, and I can see that there is definitely a good apoptosis of the cells has happened. The moment I have given mefloquine, definitely again I could see there is a death and everything is happening. But the moment I have combined these two drugs together, I can see that the death or the survival of the cells is really affected. So what I can conclude from this particular slide is that the moment I'm going to go for a combination, established anti-cancer agent and a drug repurposed agent, right? It is going to potentiate the anti-cancer activity, which can talk about like it can decrease the the side effects that is caused by the anti-cancer agent. So some of the good points: the drug repurposing potential of mefloquine was delineated. Mefloquine potentiated the anti-cancer activity of cisplatin in a breast cancer cell line. Now the similar work is also going in cervical cancer cell line. Once all my in vitro data is going to complete, I'm having some collaboration with the ACTREC so that I can go ahead with some in vivo studies and all. Such combination therapies can be economically feasible and also can decrease the toxicity of the chemotherapeutic drugs. Similar kind of a work. So we are working on this antiviral ritonavir, which was again proposed to treat, SARS-CoV-2. There was one very interesting drug which we have got a very interesting results, dexipeprazole. Like being an antipsychotic drug, this drug has shown wonders, in the case of a breast cancer. So we're trying to delineate the entire property, entire mechanisms and all. There's one more drug called as a mislazine. Again, it has again showed some very good promising result. There's one more PG student is working on ripaglindine. She has just started with her. She's into the all molecular modeling, drug repurposing to her, and we're thinking of going ahead with this particular drug. So this is my team at DY Patil University. These are my students. Without the students, it is definitely not possible to present any kind of a work. Shrikant and Tandon were the one who has, they were the stepping stones of this particular, where we have started this particular project. Some of my projects also deals with the nanotechnology. So Shada was working on the nanotechnological aspects of some other products. Dayanand, Priyanka, Aishwarya, Yash, these all have contributed a lot to my drug repurposing thing. I would like to acknowledge Dr. Devjani Das Gupta. She has provided me all the facilities. Dr. Tankamani Mara ma'am, she's the Dean Faculty of the Science. Some of my, pillars of my support in my department, these are Dr. Kanchan Leta, Shinoba, Muslim Saburi, and definitely the other faculties of SBB. So with this, I would like to stop sharing my screen.
Yeah, thank you, ma'am, so much for the wonderful talk, and it was very insightful on about drug repurposing and everything.
Yes.
So moving ahead,
thank you so much.
Yeah, our second speaker would be Dr. Shashi Kumar S, scientist at Avesta Gen Private Limited, Bangalore. I welcome, sir.
Hello.
Yes, sir.
Can I audible
perfectly
all
audible.
to you?
You can go ahead.
Yeah.
Yes,
Can I share
sure,
my presentation now? Yeah. Hello.
Yes, sir. The slide is visible.
Slide is visible. Okay, thank you.
Yeah.
Good morning, everybody. Now I am going to talk about the nutraceutical and functional food for 20th century for alleviating lifestyle diseases and like obesity, diabetes, and CVD. So nowadays is very youngsters are more of going to the junk food and other foods. So these are the foods what we are now we are talking is modifying lifestyle, and also it is um, helping them in alleviating diseases like obesity, diabetes, and CVD. So next, for example, nutraceutical is a term coined by Stepan D. Fickel in 1979, and it is defined as a food or parts of food that provide medical or health benefits, including the prevention and treatment of disease. This is what the definition is. So major active nutraceutical ingredients in plants are mostly flavonoids, and these are typical phenolic compounds and antioxidants. So they also have a long recognized to possess anti-inflammatory, anti-allergic, hepatoprotective, and anti-thrombotic, antiviral, anti-carcinogenic activities. These are well known. Now I will tell you some interesting things here. Next slide. Now we go back to the 700 BC and 400 BC, where Ayurvedic Charaka Smita and Sushruta Samhita is telling. So they have identified so many plants and other food ingredients for the alleviating so many diseases. So Ayurveda is an alternative medicine system with historical roots in an Indian subcontinent, particularly in India. The theory and practice of Ayurveda is pseudo-scientific. Now, what I am telling you is a bionutrition approach which combines traditional Ayurveda and plants and plant products used are validated scientifically, and all scientific pharmaceutical protocols are used to validate these Ayurvedic data and find out the molecules that is responsible for prevention of lifestyle diseases. So what methodology that we use for scientific validation of these plants? So validating and finding biomolecules responsible for prevention of disease. And we develop a database of information from traditional system of Ayurveda and other system like Unani, and so many other systems are there where plants they particularly used for the different purposes. Not only plant, there are so many other um, molecule materials also is there. So integration of modern scientific protocols, like already Jyotirmai has told what is the pharmaceutical protocols they use to drug discovery. Same way, we also use these drug discovery protocols. So finding the molecules and integration of these molecules as a food additives, or it can be added to any food materials or daily incorporation into our dietary regime. So that is where this prevents um, diseases. So for example, we will go first as a rice as a nutraceutical. Traditional varieties such as basmati, they have a very low glycemic index, useful in weight reducing diets. Rice based ORS is also reported to be um, they are very low glycemic index content, and this is better than the glucose. So there, even WHO has recommended this ORS. Inulin, other we can talk about the controlling obesity and other things. A substitute for sugar. Inulin is a long chain oligofructose. This can be obtained from chicory, Chicorium intybus, or this can be also from the fermentation of sugar. So this long chain fatty acid oligofructose, this can be a substitute for the sugar. So when consumed, they have very low glycemic value, hence can be incorporated in variety of foods. It's a replacement for sugar. Also has acts as a prebiotic, and they lower the cholesterol and good lead for the obesity. Now we have already done a lot of research and validated plants scientifically. I will take a few examples. Already we have Gojimax, Lycium barbarum. It is for immune health. So immune health, now you know the COVID-19. So you have to improve your immune health, means you have to use a dosage of 7 to 14 grams of this whole plant, whole fruit of Gojimax. This can be added to any of our dietary food or daily food. This can be used, or that can be manufactured in processed food also. This can be added. Even it is also used in AMD, and we have given the what are the dosage and mm, bioavailability. The molecule which is zeaxanthin. Next is we talk about anti-diabetic, where we have a cinnamon xylonicum, Salacia reticulata, and Eugenia jambolena. These are the anti-diabetic. So dosage already given in the this can be added to any of our morning breakfast food or anything. So it has to be taken in a long-term basis. So prevent the diabetic condition. It may not cure the diabetic, but it is anti-diabetic. It's a preventive medicine. Next is again we have anti-diabetic Lagestromia speciosa. These are the we have given. This is what is the dosage is required for oral administration. Administered 50 mg to 1000 mg per kg of human body weight, and extract from leaves of this 1% chlorosolic acid, which is a glucosol, that is responsible for the uh, clinical trial involving type 2 diabetes. These are clinically validated. Now we also talk about the obesity, where we have the HCA, hydroxycitric acid, and it is well known actually. And these clinical studies, everything has been done in this. 1,500 mg of hydroxycitric acid per day is required to reduce the obesity over the period of time. So this is already so many medications already in the market. So that can be added into the any food additives also. Another good examples are green tea and green coffee. This is also weight management and obesity, and the dosage is given here, 200 mg per um, 200 mg per serving the green tea extract and green coffee. There are another soya influence and Aspergillus racemosus. This has been validated for human health. Women's health is a very important thing, and 5 mg kg per body weight and post-menstrual women. And most of the influence are in intact soy proteins. Diet was approximately equivalent to human dose of 140 mg per day. Adult human dose is 20 gram per day. Aspergillus racemosus. Thank you.
Thank you so much, sir, for the very amazing talk. Now, I have put in the chat box about the question. So, I guess we have a question for Dr. Jyotirmoy ma'am. So, there's a
Yes.
question by Kriti saying, how do you take care of the side effects caused by the combination of repurposing drugs and already existing anti-cancer drugs?
Yes. Yeah, I'm audible. Yes, Kirti, I think that's a very nice question. But what we are currently doing is that from the databases, from the in silico thing, we try to identify the compounds, right? We perform a molecular docking, we do extensive in silico method validation and all, and we further go for in vitro data. Now, in vitro data, it will be very difficult to identify that what would be the side effects and everything. So I think your answer is going to be very much would be addressed, like if we are going to go for an in vivo, validations and everything, because then the side effects would be much more pronounced and all. Because in vitro, when I talk about, for example, if it is a breast cancer or if it is a lung cancer, it would be just a one cell type. It's a one pure population of the cell lines that I'm going to take. I'm going to do some experiments, right? But that is not going to completely reflect whatever is going to happen inside the human body. So previously I have referred some papers and all. So these people try to, use that particular concentration of the repurposed drug, so that it is going to just simply potentiate the anti-cancer drug, and it is not going to do any other, side effect of its own. That is the thing. So again, for example, if there is a particular dose for any diabetes, or it would be for leprosy, now the same dose won't be useful for the anti-cancer. It is not that he was same dose he use hoga. It would be a different, dosage depending upon your in vitro, in vivo, your phase 1, phase 2 clinical trials. It is going to get decided. Right, Kirti? I
Okay.
hope I've answered. Thank you so much. Thank you.
Thank you,
May I
ma'am.
ask one question?
No, you can put in the chat
May I
box.
ask?
I'll be moderating the session. You can put it in the chat box.
Okay.
All questions can be put up in the chat box.
Yeah.
Yeah. So so the second question by Ashwini Kumar is for Dr. Shashi Kumar sir. Isn't excessive antioxidant as shown with green tea example harmful to humans?
No, actually, the dosage can be adjusted based on the body weight and kg of body weight. So that can be clinically validated. So that is actually the clinical validated dosage what we have shown.
Okay. Okay. Thank you, sir. And then
Thank
we have one more question for Jyotirmoyi ma'am.
Yes, yes.
Can the combination of drugs be used to somewhat help in tackling viruses such as HIV and AIDS by Subrat?
Yes. Yes, Subrat, I think again it's a very good question, and there's a combination of the drugs has been used for the HIV and AIDS as well, like ritonavir and lopanavir. I think they are the traditional antiviral disease. Probably, I feel that since I have been working for last 2-3 years on this particular topic, I feel that the drugs, they do have a good repurposing potential. We just need to explore them and again go for the further assays and all. Right? It can be definitely be done. Okay, I
Okay.
think one more question by Himanshu. He has asked, like, after
Yeah.
docking studies, what steps we have to follow for further analysis? Yes, Himanshu, I think after the docking, once we have done, what we do is that we go for a particular, we choose a breast cancer model or a lung cancer model, depending upon, where you are expecting that your drug is going to show a good result. Then once you have chosen your in vitro model, then we do a series of assays. Starting the first assay that we do is an NTT, because we need to know like which all concentration of both the drugs are going to give a synergistic effect. And then after that, there is a huge lot of a combination studies that we do. We do analysis by a Compune software that is going to tell me like which all combinations are synergistic, which all combinations are antagonistic, right? And those combinations are further taken, and I do a lot of functional assays. Like I do my wound healing assay or a migration assay or a clonogenic assay. I need to perform a lot of functional assays just to show that, okay, whatever repurposed drug I have taken, it would be definitely, it is going to, potentiate, or it is going to improve the efficacy. Basically, it is going to work as an adjuvant. I think, Himanshu, we can work upon this. You can just contact me, and again, after the summit, we can have a discussion. So basically, that's the purpose of the summit, to have a collaborations, to have some discussions.
Okay, so we have one more question by
Yes, Parul.
Parul.
Parul, yes, Parul, I have worked upon lung cancer cells as well, breast cancer and cervical cells, cancer cell. All the drugs are different, and wherever I see a good repurposing potential, I choose my model accordingly. Okay, are you 100% sure COVID-19 is not a bacterial disease? It's a truly a viral disease. I think, yeah, I think it's been published. It's been again reported that definitely it's a viral disease.
Okay, so I guess we are done with the questions as of now.
Yeah.
Sir,
Thank you, Aditya.
thank you so much. Yeah, thank you so much, ma'am. Thank you so much, sir, for for the wonderful talk. We'll now go ahead. So our next speaker, actually, Dr. Prasen Binchurkar, Director of Research and Development, Red Carbon Techno Hub Pune, unfortunately due to some network issues is not able to present. So we'll be moving on to our next honorable speaker, which would be Dr. Shilpi Sharma, ma'am, Professor at Savitribai Phule, Pune University, Pune. I welcome you, ma'am.
Thank you so much. I'll just start sharing my slides.
Yeah, sure, ma'am.
So I'm hoping my slides are visible.
1 second. Yeah, now it's loading. Yes, yes, the slide is visible.
Okay, so thank you so much. I would really like to thank the organizers for giving me an opportunity to present my work here. My lab basically works on diabetes, and today's talk would be focused on the potential of using the human islet amyloid polypeptide as a target for type 2 diabetes. As the moderator has already told, I'm working as an assistant professor in the Department of Biotechnology, Savitribai Phule Pune University, and my lab there focuses on the research which we'll be talking about today. So, one second. Okay, is my pointer visible? Can you see my pointer?
Yes, ma'am.
Okay. All right. So just to introduce everybody to diabetes, diabetes mellitus is a disease which was initially considered to be a disease of the West, but now it has been seen growing in increasing numbers in the developing countries as well. According to the reports of the American Diabetes Association, more than 420 million adults have diabetes. And the problem which is now coming up is that the disease is majorly affecting adolescents and young adults. So initially considered as the disease of the West and of the elderly people, we are seeing it getting developed in adolescents and young adults also. The problem why this is happening is, first of all, there are different reasons for that for each country. For India, we have changed our lifestyles altogether. We are following the West in terms of the lifestyle. We have a sedentary lifestyle with no exercise. We've changed our food habits. And the other problem is that there is a very bad, I should not say that. The biomarkers which are available for diagnosing diabetes are not that reliable. So basically, you rely on the plasma glucose levels, or you do an oral glucose tolerance test to see whether a person is having diabetes symptoms or not. But those are not disease symptoms, tests which will tell you that diabetes is developing. It will tell you an indication once the disease has already set in the system. And as everybody would know, it is very difficult to reverse diabetes. Once it is set up in the system, I don't think many drugs are there which are able to reverse diabetes altogether. They only act in, trying to suppress the symptoms. So I'll just, introduce everybody to diabetes. So my lab basically focuses on type 2 diabetes. So what happens, so this is the pancreas in a healthy individual, and these are the peripheral tissues. So what happens is when you take a meal, the glucose levels rise in the peripheral blood. As a result of that, insulin is secreted from the healthy pancreas. It goes and binds to the insulin receptors, as a result of which glucose, the receptor which is involved in the gets to the membrane, as a result of which glucose gets internalized in these cells, right? So this is how glucose metabolism occurs, and then it is used for the downstream processes. Now, there are two major types of diabetes which are present. Type 1 diabetes, it's an autoimmune disease which occurs in generally very young children, and it's actually associated with the destruction of the pancreatic islets of Langerhans, as a result of which no insulin is produced. The glucose remains outside. No glut for membrane localization occurs. As a result, you get hyperglycemia. So when I say hyperglycemia, it means excessive glucose concentration in the body fluids. Now, we work on type 2 diabetes. And what is type 2 diabetes about? Type 2 diabetes occurs when the peripheral tissues stop reacting to the insulin that is secreted. So after a meal, the insulin gets released from the pancreas. However, it is not able to go and occupy the insulin receptors, and as a result of which glucose translocation to the membrane does not take place and glucose internalization does not occur. So insulin resistance is one major feature which is linked with hyperglycemia, which develops in type 2 diabetes. Now, there's another field of thought which says that in addition to the insulin resistance being developed, the pancreatic islets of type 2 diabetics do not produce insulin in the amount that they should produce because they start dysfunctioning. They do not function at the level they're supposed to. And due to the constant insults which happen in the system, they start undergoing apoptosis or they start dying. So my lab basically is, we are trying to look and understand diabetes, and there are different thrust areas which are there in the lab. We do biomarker identification using NMR-based metabolomics. We also try to look at the non-coding RNAs and how they are involved in causing diabetes. There is another field of thought where it is thought that instead of the beta cells dying and leading to the causation of type 2 diabetes, they actually de-differentiate into another phenotype. They either can de-differentiate or they can trans-differentiate into another islet lineage, or they can go to the progenitor lineage. So I'll not be talking about these three thrust areas, and today we'll be focusing on the human islet amyloid polypeptide mediated cytotoxicity in type 2 diabetes. So just to introduce you to the human islet amyloid polypeptide, it's a glucoregulatory hormone, and this is the hormone which tells you that you need to stop eating. You've done enough. So it's a satiety-inducing hormone, which is produced by the pancreas. Now, if I say it is produced by the pancreas, I mean that it is co-secreted and co-produced with insulin. So this HIPP is also referred to as amylin. A lot of people call it as amylin. So the amylin to insulin ratios are 1:100 in a normal individual. So when I say this, in terms of the production, if 100 molecules of insulin are being produced, 1 molecule of myelin would be produced. Now, what happens in hyperglycemic conditions, which are associated with diabetes? Insulin content increases as a result in the initial phases, which results in hyperinsulinemia. Now, the cells are able to manage insulin properly, but because amylin turns to be an amyloidogenic hormone, it tends to get misfolded. It self-associates and forms amyloid deposits and plaques. So if you look at this figure here, the A, B, and C panels are from the human pancreata, and you can see that these red colored deposits are the Congo red stained amyloid deposits which you see. Here you again see the deposits in these pancreata, and these are the electron micrographs of the human pancreata. Now, the problem with this hormone is that there is a very big species difference in this hormone, right? So the human IAPP or the HIPP, which we work on, tends to misfold and form aggregates. On the other hand, the hormone which is produced from in the rats or in the mouse models, which are actually the first animal models which we go to for performing these studies, there is a sequence variation. There are a lot of proteins in the sequence which do not allow this peptide to misfold and form these aggregates. So these are the D, E, and F panels are images which are taken from the pancreata, isolated from HIAPP transgenic mice. So in here, what we have done is, instead of the rodent IAPP, we have the human IAPP being expressed, and these are at different ages. So this is of a young mice. The panel T depicts the pancreata from a young mice. As they age, as is seen is in E and F, you can see the intensity of the green stain increase. So you can see that the tendency of these mice to form these aggregates, which are stained with thioflavin S, which is a stain that goes and binds to the amyloidogenic aggregates, increases. So you can see the entire pancreas actually is filled with these islets, as a result of which these pancreas do not function as well. Okay, so now there is so most of the work which has been done for this hormone has been done either in the transgenic mice, or it has been done with in vitro models. And even in the in vitro models, most of the studies have been done using biophysical work. Very little cell mediated work has been done because of the problems which I'll talk about in the next slide. So we'll first talk about the HIPP aggregation. So what happens is, once it is produced in normal concentrations, it maintains itself as a monomer. Now, but when it tends to get overproduced with insulin in hyperglycemic conditions, as is seen in type 2 diabetes, what happens is that these tend to form these monomers, they tend to form oligomers, which are again soluble. And then these oligomers go on further to form protofibrils and fibrils. So it is these protofibrils and fibrils which are going to be seen as those amyloidogenic aggregates, as I've shown you in the previous slide. And they are the ones which are going to cause cell death. So once these fibrils and protofibrils keep increasing in the pancreatic islets, it is going to result in cytotoxicity. And it can happen with seeding, it can happen without seeding. If it happens with seeding, the tendency to form these aggregates is much higher and much earlier as compared to when it happens without seeding. Now, I told you that there is a problem with this hormone, that you cannot use the normal, models of diabetes for doing these studies, because those animals will produce the rodent IAPP, which is not going to aggregate, which is not going to misfold, and as a result, they end up being useless. The other problem is because it's only a 37 amino acid long peptide, to be very frank with you, but when you try to produce it using synthetic methods, it is very difficult to produce it, because it ends up being, you do not ever get a good yield. It becomes very expensive, and there's a lot of problem associated with it. So our lab, in collaboration with Dr. Ashutosh Kumar from IIT Bombay, we developed a protocol for producing recombinant IAPP. So we also filed a Indian patent for that. So this is your mature IAPP, which we purify first as a fusion protein, and then in the end you see that there is this peptide which elutes out, and it's the correct molecular weight which we expect for the peptide. So we've been the first ones who have been able to produce this peptide in a large amount in the lab. And the other important part which I would like to emphasize on is that in addition to producing it in large amount, we have been able to produce a functional peptide, because people who do claim for producing this peptide before us, the peptide was either produced in very little quantities, or it was not functional at all. When I say functional, I mean that it was not inducing toxicity or cell death in the pancreatic cells which they were using for the analysis. Okay, so just to move forward, we wanted to know, because I told you before also that there are very few reports about this peptide in cells, because we don't have a model system for that. So Professor Chris Wallum and Chris Mackler were very kind enough to give us these INS1E cells. It's a pancreatic insulinoma which is produced from the rat. And the good point about these cells is that they respond by producing different amount of insulin to the different concentration of glucose in the extracellular media. So if I change the glucose concentration by even 1 millimolar or 0.5 millimolar or 5 millimolar, the amount of insulin that is going to be released from these cells is going to be very different. Okay, so we wanted to check whether this peptide that we have produced using the recombinant methods is actually inducing any cytotoxicity, and how it is inducing this cytotoxicity. So if you can look at these, it's a really busy slide, and I'm sorry for that, but because I wanted to point out a few things, so I've kept it like this. So as you can see from the panel A, the cells start dying when you add this peptide in them. Right, so it's a 24-hour experiment incubation with the peptide, and you see that there is a tremendous loss of viability once you expose these cells to IAPP. Now, the panel B tells you the staining for annexin 5 and propydiamidite. These are stains which help you distinguish between apoptotic versus dead cells. So if you look at this panel here, the upper right panel, this is the panel which shows you the cells which are undergoing apoptosis and which are in the late phases of the apoptosis. So when we quantified these cells, we found that the amount of apoptosis was tremendously high in cells which are exposed to 2.4 micromolar IAPP as compared to the control cells. Not only that, next we found out how is this cell death happening. So we measured the mitochondrial and cellular ROS levels in these cells, and we found out that these levels were very high in cells which were treated with IAPP. Also, there was a lot of membrane damage, which was assessed by the T-bars assay. Along with that, because oxidative stress induces DNA damage, we went on to check for the DNA damage marker, which was gamma H2X. And you can see there's a lot of green color lighting up here in these cells, which were treated with IAPP. And these are the cells which are the control cells, and you do not see much green here. So that means when you add IAPP to the cells externally, these cells start undergoing apoptosis because of the presence of DNA double strand break being formed. We also looked for other markers for apoptosis and oxidative stress. And as you can see from this G panel, there was increase in oxidative stress as well as there was increased apoptosis. Now, because we were working with a peptide, we had to show that it is affecting the pancreatic cells, right? So when we tried to publish the paper, the first question that the reviewer asked was, are these affecting the phenotype, the functionality of the islets, right? So we did not have functional islets in this at that time, because insulin cells at that time we were growing as a 2D model, right? So what we did was we isolated the islets from the mice, and we exposed these islets to the IAPP, and this is the insulin secretion that you see from these islets, right? So this is the control scenario in panel H. You will see that in basal glucose concentrations, that means at glucose concentrations which are equivalent to 100 to 110 milligrams per deciliter in the human plasma, which is for a normal individual. You see that there is some amount of insulin secretion that happens. And when you stimulate these islets with high glucose, which is like 16 millimolar, which is like equivalent to 250 milligrams per deciliter after a glucose-rich diet or a carbohydrate-rich diet, you do see that there is a significant difference between these two. But what happens when you add IAPP to them? There is a significant decline in the stimulated glucose release. That means these islets are no longer functional like before, and they are actually not releasing insulin at the required concentrations as they should under a high glucose concentration. So to summarize this, I can easily say that this IAPP actually forms aggregates, and these aggregates mediate apoptosis, mitochondrial dysfunction, they disrupt the cell membranes, and I've not talked about the other things here, but there are a lot of ways in which these peptides actually mediate cell dysfunction. Now, before me, I would like to thank Professor Sashi Kumar for bringing up the anti-diabetic natural compounds that he talked about. So there have been a lot of recommendations by even the American Diabetes Association to include a lot of Indian spices and Indian food supplements which we take, like neem, tulsi, cinnamon, for just as supplements to the drugs that you take for diabetes, right? And what people have been finding out using different methods is that these natural compounds actually help in reducing the blood glucose levels. So my lab actually wanted to check whether these natural compounds actually act as inhibitors of IAPP aggregation or not, and we worked on natural compounds isolated from neem and from jamun. But for the talk today, I would just be focusing on azadirachtin, which is actually a lemanoid, which is extracted from the neem plant. Okay, so okay, so just bear with me. So before we go on to the cellular assays, it's very much required that you give a in vitro proof of principle that actually this compound is working in the direction in which you are thinking. So I told you there have been a lot of biophysical studies. So taking leads from those biophysical studies, we also performed some biophysical studies in the lab. And this panel A gives you the thioflavin T efflorescence intensity. Now thioflavin T, like thioflavin S, which I showed you in the first slide, where we could see the green color come up, is something which goes and binds to amyloidogenic aggregates, which the HIPP forms. So if you can look at the blue line here, this indicates HIPP. So you can see that HIPP aggregation starts at as little as 2 hours and it goes on till 12 hours. Right, but if you add azadirachtin into these HIPP peptides, you do not see that much amount of aggregation. This is another way of identifying the aggregation potential. And you see that in contrast to HIPP, when you add azadirachtin into the formulation, you do not see that much amount of aggregation. Now, these are the the TEM and the AFM images, and you can see that HIPP forms very nice fibrils. You can see long elongated fibrils, but when you add azadirachtin in either molar ratios of 1:1 or 1:5, these fibrils break into smaller aggregates. Okay, you do not see those fibrils anymore in the last two parts of panels E and F. Right. Next, we wanted to check whether is it able to relieve the cytotoxicity associated with HIPP. So we did these cell viability assays, and as you can see here, there was a lot of cell death in HIPP. But when we added azadirachtin, the cells could come back to normal. They were able, the viability was restored. Not only that, the increase in the cellular ROS levels, which we see, are actually coming down to normal when you add azadirachtin to it. So that means azadirachtin has an anti-amyloidogenic potential, and it has a potential to prevent HIPP-mediated cytotoxicity in the pancreatic INSWANI cells that we have used as a model system here. So this was to see whether it is preventing aggregation or not. Now, when I started, I said that diabetes is a disease which is not reversible. So the next thing that we thought about was, is this compound, is this natural product able to act on preformed aggregates? That means, if at all the aggregates are already formed in the system, is azadirachtin able to break them? So as you can see from these thioflavin T experiments, definitely we saw loss of fluorescence intensities. Even in the TEM and the AFM images, you see that the preformed aggregates which were there from HIPP were broken down when you added azadirachtin to it. Okay, now again, cell viability was restored and the ROS levels were also restored. So we next wanted to find out whether, what is the mechanism by which it is acting. So I told you the the cell death was mediated through apoptosis, and azadirachtin was able to restore or reduce the apoptotic cell number in these cells. Also, we did an assay where we went on to measure whether it is affecting the cell cycle, right? Because most of the time it is seen that apoptosis happens because these cells get stuck in some phase of the cell cycle. And as you can see here, the INSWANI cells, if you just look at the blue color, the cyan colored bar, they have a very good G0 phase, G1 followed by an S2 and a G2M phase. But when you add HIPP, you see a decline in the G1 phase and the G0 cells increase. But when you add back azadirachtin, you see a restoration in the cell cycle profile. So that means the cell cycle inhibition which was observed because of the HIPP, that has been able to be restored because of this. The membrane damage was restored, and one important thing. So everybody knows about mitochondria, right? Mitochondria plays a very important role in restoring cellular function, right? So the addition of HIPP was associated with decline in mitochondrial membrane potential. You see this cyan color peak shifting towards the right, but when you added HIPP, it was restored. Again, the insulin release from the pancreatic islets, these were clean experiments done in mice, the islets is rated on mice. You see that HIPP declined the insulin secretion, but when you added azadirachtin to it, it was able to bring it back to normal. So next thing was, how is this working? So we went about, and thanks to Jyotirmoy for explaining molecular dynamic studies in such detail, so I don't have to do that part now. So basically, I'll just summarize this slide in 2 lines that HIPP oligomer is proposed as a pentamer. That means 5 molecules of IPP stack on one another to form an aggregate. And so for that purpose, we added 5 molecules of azadirachtin in the MD studies, and we could find that there were different sites recognized by azadirachtin on the HIPP pentamer, and the twist angles also varied a lot. So these results, so to conclude so far, we can say that we were able to come up with a novel protocol for the synthesis of human islet amyloid polypeptide using recombinant DNA technology. This peptide was able to mediate cytotoxicity and dysfunction in the pancreatic beta cells and islets. And azadirachtin, a secondary metabolite isolated from the neem plant, which could restore the dysfunction and provide protection from the HIPP-mediated cytotoxicity. So the next, I'll just take 2 more minutes. The next question which came into our mind was, these experiments which we did before were for a 24-hour exposure. So we wanted to know how bad is bad. So how little an exposure of HIPP could induce problems, and I would not go into the details. We did a time point and a concentration dependent experiment, and we found that as little as zero point 0.3 micromolar, which is like 1/4 of the IC50 concentration, we could see problems of oxidative stress, cell death in these cells, and even with the exposure of 1 hour. Next, how little a time is it required for the amyloidogenic aggregates to form? And we found that as little as 6 hours, as if you could see at this panel here, was enough to induce the aggregation process. And this green color increases at 24 hours and 48 hours. So we are further in the process of doing investigations on the metabolomics front, where we're identifying what are the metabolites which are perturbed in these conditions. And Shraddha, one of my students, is going to be presenting that as a poster. So to acknowledge, so I've been very lucky to be working with very hard-working students. Devika, my first PhD student, was involved in standardization of most of the experiments. Pratiksha and Shraddha work on HIPP-mediated cytotoxicity. Nikhil was a MSc dissertation student, and this is the lab alumni, which constitutes mostly of MSc dissertation students. These are my collaborators. No work is possible without collaboration and funding. So we collaborate with a lot of people, and funding from DBT, UGC, and Savitribai Phule Pune University is acknowledged. We've also been lucky to get a grant from the Vadhwani Research Foundation, IIT Bombay, for this HIPP project. And in the end, I would just like to advertise one of the books that I've been involved in editing. It's called as Prognostic Epigenetics. And if you want, you could just have a look at that. And I'll stop sharing my slides. And if there are any questions, I'll be happy to take that.
Yeah, thank you so much, ma'am. So we have two questions, I guess, two important questions. One by Ashwini saying that HIAPP perhaps coexist with insulin in the same granule. Is there any chance misfolding in HIAPP can also include distortion of insulin?
So insulin distortion also occurs. Insulin also has a tendency to form amyloidogenic aggregates, but the potential of HIPP to form amyloidogenic aggregates is much higher than that of insulin. So insulin does not cause that big of a problem because of aggregation as HIPP does. So I hope I answered the question.
Yeah. So there's one more. Is it possible to control the pain of diabetes? Is it possible to control pain of diabetes in patients with physical methods? For example, TENS. Is there a physical way to treat pancreatic secretions defect? I'm interested in that area by Iyad.
If I understand the question right, endoplasmic reticulum he's targeting to, and he wants to ask whether ER dysfunction can be corrected, right? Is it okay? Is that
what the question tells, I guess? Yes.
Yeah. So yes. So basically, endoplasmic reticulum is involved in removing the misfolding proteins, right? So basically, people have been trying to work with different chaperones, which are able to prevent the diabetic conditions because of endoplasmic reticulum defects. So yes, there are a lot of people who are working in that field. I did not present the data here, but we also have some very good results with the EF2 alpha and GRP78, which are involved in the ER conditions.
Okay. So yeah, one last question by Dr. Shashi Kumar itself. Need to know azardictin is used as a biopesticide. Have checked it as anti-diabetic in human. Have you done the toxicity studies, ma'am?
So azardictin was initially used as a pesticide because of its the kind of oxidative stress that it generated in these insects, in this pest, and mediated their cell death. So we are using AZA at a very low dose. We are using it at a dose that it is not causing any kind of cytotoxicity in the cells. And preliminary results from what we have done in the mice system also shows that it is not that problematic. So very little amount we are using. We're not using it at concentrations. So definitely, if you increase the amount, it does cause death. So the amount that we've used here is not inducing any cytotoxicity in the cells.
Okay, thank you so much, ma'am. Then
it's a pleasure. Yeah.
Yeah, so now moving ahead, our next speaker, that is the fourth speaker for the day, would be Dr. Praveen Poddar, HOD Molecular Medicine and Stem Cell Biology, Mumbai. So I welcome you, sir. Sir, yes.
Okay. Should I start?
Yeah, you can start, sir.
Okay. Hello, can you?
Yes, your screen is visible now, sir. So you can start presenting the PPT.
But I can't see the PDP. You can see the screen.
Yeah, we can see the screen, but you have to
Screen
start
is, but
presenting the PPT.
acha, okay. So how to go about that?
Sir, you have to click your. Have you the presentation, right? You might have the presentation. So
Yeah, on
just
the
click the
desktop. 1 minute.
present.
Just
Yeah.
1 minute.
Yes. So you can make it on full
Can you?
screen now. Yes, full
Yeah.
screen. It went away, sir.
Just 1 minute.
Yes, it came
Is
now.
there
Yeah, it is there now. Just make it full screen.
full
Yeah,
screen?
down there is an option, right? Yes,
Yeah,
that's.
I know that. I know that. Okay.
Yeah, now it's visible. Yes.
Now it is visible. Okay. So, but I can't see you people. Is it okay?
Yes, no problem, sir. You
Okay.
it's a feature of Google Meet. You will be only able to
No.
see the
Yeah.
presentation.
No problem. Okay. Good afternoon, everybody. I am Dr. Praveen Poddar. I was a former head for Department of Molecular Medicine and Biology, Jasmine Hospital Research Center, Mumbai, for last almost 12 years. Before starting my talk, I would like to thank JAX Foundation and Deepa Patil and Tanaya for giving me this opportunity to talk in this uh, summit. I will be talking mainly on the technological evaluation has been taken. My title is Innovation in Medical Biotechnology, latest update on innovation in technology and therapy. So I will be mainly highlighting the revelation made by the various scientists, Nobel laureate, to develop this molecular medicine field. And first, it is my duty that to salute the Nobel laureate of 2020, for Dr. Harvey Alter, who is from NIH. I am also a fellow of NIH, so I am very proud of him. He has been working to NIH for many years. He is a very old person, and he has been working on hepatitis for several years, and he is the first person who has initiated and shown that the hepatitis C is involved in hepatocellular carcinoma as well as the cirrhosis of liver. Along with his Hoxton and Dr. Rice also had joined to give him the support to work on this hepatitis C, which is one of the really pioneer area in the field of molecular medicine. I also involved a lot of work in this area. The second prize in chemistry, this is a prize in physiology and medicine, where the Nobel Prize in chemistry has been gone to the very young ladies. One is Jennifer Dondra and Emmanuel Charpentier. They have done extensive work on CRISPR-Cas9, which is a new technology. Last 5 years, I think we are listening about it, and it is a technology which is a gene editing technology, which will really help us in doing several recombinant technology or making the clones or deleting or incorporating genes, and this is going to be very easier for us in near future. So these all people have really helping us to get a better innovation in our technological background. Just to give my brief idea about the international organization associated with my scientific activity, actually I have done my PhD from Tata Memorial Center, Mumbai. After that, I had a chance to go to NIH, National Institute of Health, where in 1995, I have cloned 3 novel genes in my postdoc work. These are GTTRL, KPL1, KPL2, they are on database. After that, I have not have intention to stay in US, but I come back to India, work more few years to Tata Memorial Center, and then I left for US again, where I got a faculty position at MD Anderson Cancer Center, which is one of the topmost cancer center in the world. And I had took that opportunity. I worked there for 3 years, work on molecular targeted therapy in prostate and pancreatic cancer. I also had a really great experience with visiting Cold Spring Harbor of James Watson Laboratory, which is really my golden days for that time. So after coming back from MD Anderson, I joined Department of Molecular Medicine as a head at Jaslow Hospital, where I found the lab was well equipped for doing the molecular medicine work, like a real PCR, real-time PCR, sequencing, all these basic instruments were available for. So I have really taken a chance of that and established a well-functional molecular diagnostic and stem cell research laboratory, where I have trained more than 1,000 students in this area, and they are already working in various universities in US, UAE, UK, some in Harvard, some in Stanford. So these are really good credentials for me too. I have worked with some IIT students also in Roorkee, and they have published several papers. Recently, one paper has been published. Coming to our main topic of innovation in technology, the main hero involved in making revolution in the field of molecular medicines are the many of the Nobel laureates. James Watson and Crick is first in 1953. He has first shown that it is a double helix. After 10 years, he got a Nobel Prize in physiology, the discovery concerning molecular structure of nucleic acid and its significance for information transformation in living material. Next prize goes to the very important step in our polymerase chain reaction is making the synthesis of oligonucleotides. We are really fortunate that one of the Indian American was involved in this Nobel Prize is Har Govind Khorana in 1968, Nobel Prize in Physiology or Medicine. Then the steps has been come that how we can amplify the DNA. That work has been done by Carey Mullis and Mackenzie Gold. He got the Nobel Prize in 1993 in chemistry for invention in polymerase chain reaction. He has made the first prototype of the PCR machine, which presently we are using very easily. And this is a machine, is mainly the heart of all the molecular biology. Another thing, 1976, again, discovery of Tech polymerase has evolutionized this polymerase chain reaction. The other person who is a Sanger sequencing, we know the Sanger sequencing, which has been done by the first discovered by the Sanger, who got a Nobel Prize in 1958 for elucidating insulin protein structure. He also got the Nobel Prize in 1980, along with the Paul Birch, Walter Gilbert, for the recombinant DNA technology. So these are all Nobel laureates had really made the lot of great contribution in developing this molecular medicine field. So I am going to talk about the introduction to molecular diagnostic and health diseases. Initially, what I want to tell, because this is mainly the student made the, I thought that it is most of the postdoc students or postgraduate students are attending my seminar. So I thought that I should give a brief idea of the technology involved in this molecular medicine. Molecular medicine has been launched in 25 years ago. Molecular medicine inspired to collaborate across a basic clinical discipline to accelerate the fundamental knowledge and advances for detection, prevention, and treatment of human disease. Molecular medicine enabling scientists and clinicians to fasten the translational discoveries of human suffering from diagnosis to research, diagnosis to reach the precise theory that we can call as a bedside to bedside. Because doing only basic research When I was in Cancer Research Institute, Tata Memorial Centers, we were just making the work in the test tube. We were never thought that how this will go to the patient. But when I came to Just Hospital, I have selected Just Hospital because I wanted to have some correlations with the patient's health, disease. So there I could got a chance to develop a molecular diagnostic laboratory. We have done several work in this area. So that I will be highlighting at the end of the talk. So the molecular diagnostic has been mainly initiated by the incorporation of, means the discovery of completion of the Human Genome Project by NIH Director Francis Collins in 2003. He has published this paper in Science. Once he has declared that the Human Genome Project has been completed, there are so many companies have got a ready-made sequences which they have correlated very well with the disease, and they started developing the IVD kits for diagnosis of various diseases. The major important factor of Human Genome Project is we are all 99.9% similar in our DNA. Most of them. Individual varies by only 0.1%. That variation may correlate to a different response to the medicine and magnitude of disease risk. So molecular diagnostic is the branch which has been initiated. The more importance and specialty of molecular diagnostic is it requires small amount of blood, plasma or tissue. This is a very important. We don't need to have large amount of blood or to do lot of work. It is a very simple program. Program essay, highly sensitive and precise. Once we got positive results by any one of the sample, that means it must be a positive. It cannot be a negative again. So false positivity is very limited. If it is there, there is some problem in the assay. Accurate diagnosis, very useful in diagnosis and therapy of cancer and other disease. The other factor which I want to emphasize here is that it is always used for diagnosis as well as the therapy of various cancer, infectious disease, various things we can do with the different development of assay that I am coming to ahead. So there are different technology has been involved and how the technology has gone. So major technology are PCR, polymerase chain reaction. Everyone knows DNA, RNA, polymerase chain reaction. Real-time PCR, microanalysis. Then further in DNA sequencing, genotyping, mutational studies. Next, now it is coming to the next generation sequencing, fluorescence in situ hybridization technology, and now it is coming CRISPR gene editing technology. So coming to the technological points, we are mainly focused on the three major molecules in molecular analysis: DNA, RNA, and protein. But these machines are mainly work for the DNA, RNA assays. Now, what is important? When you are working on the DNA or RNA, the main template of your assay should be very perfect. It should be properly purified. If there is a problem in the template, the further assay will go wrong. And when you will come to know that at the end of the essay, you are not going to get any results, that time you will feel that there is something wrong. So better careful about working in this extraction of DNA or extraction of RNA. These are very important technology. I'm not going to go to the technological details here because there is no much time, but anybody wants to discuss about this or any group wants to discuss it, they can call me on my number and I can definitely help them about that. So phenolchloroform method is a usual method. There are some column methods. Coagents are made very fantastic columns, and I am very satisfied and I can recommend that this extraction also will be very useful for the molecular diagnostic where the precision is very important. Coming to the RNA extraction, still I think there is a lot of problem for various laboratories doing RNA extraction, and there are several columns are there, but I somehow I have more preferable to the triazole method extraction, which I have established in my laboratory, and we all of the time we got a fantastic results of RNA by doing taking this triazole method. There are two conformations of RNA extraction, which you have to do if you are doing work on RNA. You have to see that 260 and 280 ratio should be 1.6 to 1.2, 1.8 between. Similarly, if you want to go further purification of RNA and if you want to use RNA for NGS or any special sequencing base, you have to load this on the formamide gels where you should get 28 and 18S band. If you are getting the middle peak that you can see here, this is a degraded RNA and this is no use for using this. Now, several times what happen, RNA, actually I prefer the RNA samples should be fresh immediately, but sometimes there is some conditions we can't use RNA immediately or we have to transport. There is like RNA letter solution has been made by Invitrogen. Now that it is a Thermo Fisher, I think. There may be many other companies also may have made, which preserve the RNA and we can extract that after some days also. So these are the basic things. Now coming to the polymerase chain reaction, I think everybody knows about polymerase chain. Every student is working in this set. And but most important thing, every student has to see that they should get a proper, very precise band of your reaction. Otherwise, if you are getting some interfering bands, you cannot say your PCR is okay. It is not right. There are major 4 steps are involved: denaturing, annealing, extension, and amplification. PCR mainly amplifies, gives the million copies of the DNA, which we load on the gel and we get such type of band. This is a totally a qualitative analysis, which gives us the information that this particular gene is expressed in this particular system or particular gene is down-regulated in this specific system. It is not giving you the quantitation. For getting the quantitation, there is another method, another technology is there that I'm telling here also. Now, when we are taking RNA cannot be amplified directly. You have to make cDNA by using the reverse transcriptase, and that cDNA we have to further amplify that particular procedure called RT-PCR. So many times I have seen, I was working in Jasper Hospital, so several consultants used to come PCR, they used to say PCR, RT-PCR, all confusion, you know. So when you are working on only on DNA, we can do the PCR. When you are working on the RNA, it is a RT-PCR because RNA has to be reverse transcriptase to cDNA and then further for the amplification. So when you want a quantitation of the real, that is called real-time PCR or qPCR. This term is also many times confusing. Which is the one is sensitive? Definitely, real-time PCR is more sensitive than ordinary PCR, because in this PCR we use the technology where we label the probe, and according to the excess of reactions, the plot is formed and you get a CT value. Now I think because of the COVID, people are knowing what is the CT and all these things. But CT is not only that, you have to really calculate the viral load of this, and this will give exactly quantitation. If I am doing real-time PCR for hepatitis C or something, I can tell these patients have about 10,000 viruses. So this is a totally quantitative PCR. Is always quantitative. It is an accurate and sensitive method for quantifying the abundance of target DNA sequences. Analysis and the application of curve allow samples to quantify via standard curve. You need to run the standard curve that I have shown here. So you will get such type of peaks, and then you have to calculate you have to take a standard curve, and accordingly you have to calculate the viral load of that. Then only you are completing your assay. Presently, what I am seeing in COVID system is that they are only giving CT value. What is the load? Because this is essential, because this viral load, the treatment is depend on the viral load. And if viral load is not responding to the treatment, that means your treatment is not responding. So this is the main aim of this real-time PCR for infectious disease. So mainly this has played major role in infectious disease, and especially in the COVID sense also. So they need to make a real-time PCR quantitative analysis, not the qualitative analysis, because qualitative analysis not giving really exact copy number, and therefore the treatment cannot be monitored. So coming to the next technology which is developed is the automated DNA sequencing. Here we take a DNA and we can get the sequences. The, DNA sequencing refer to the method of determining the order of nucleotide bases such as adenine, guanine, cysteine, thymine in molecule of DNA. The chain termination method developed by Sanger and co-worker 1975 is a relative easy and reliable method to use by many laboratories. Automated DNA sequencing analysis permit easier and faster method for DNA sequencing. DNA sequencing technology has great help in sequencing of genome sequence project completed in year 2003. DNA sequencing of cancer genes has played a vital role in field of cancer diagnosis and therapy. It is a very important instrument in human genome sequence. But still, scientists has not stopped there. They have made the further development is the next. This is a peak you can get under automated DNA sequencer, and there are different colors. They are already mapped properly, so you have to just bioedit that sequences and see whether there is a mutation, deletion, and report accordingly. So another technology, gene expression profile by microarray analysis. This technology was little bit complicated one and could not progress much in this area because there was a lot of data the scientists was getting, and it was very difficult to validate the data. So this technology has used several players. Then they have made specific pathways of the genes. Here you can see at a time you can see several genes expression or down regulation in one chip. So this is the advantage of DNA micro DNA technology. Next generation sequencing. This is again one of the great innovation in molecular medicine, where now people are getting whole genome sequence done in 15 days time, which they took years and years to publish the DNA human genome sequences. This is a very, very important technology has now revolutionized the field of medicine, and I am very happy to say that using this technology, we can really find out several pathways of various disease, complicated disease. We can develop the therapies, we can develop the treatment protocols by using this system in various fields like cancer, infectious disease, or present COVID situation. So major applications of molecular medicine is diagnosis and therapies of cancer. I have extensively worked in cancer research. My major specialization for 30 years in cancer research. I am giving some of the work here. Then evaluation of infectious disease, diagnosis and therapy. Disease include HBV, HCV, HIV, M tuberculosis, now COVID-19. Here I have told you, I have also got a chance because I was in Jaslow Hospital. I have established the real-time PCR for infectious disease, and this really gives a great advantage of treatment protocol to the patient. So if there is a patient of liver cirrhosis or something, if SCV, HBV they have done, they can do quantitation and see that how much deep this infection is there and what treatment has to be given, and whether that treatment is responding. So there is no drug resistant develop or no side effects because of the drug. This is also one of the advantages. Perfect diagnosis of neurological disorders. Various neurological disorders also not been properly diagnosed. People, even I have got a patients who are actually diabetes and taking insulin. Because of the getting insulin, his all nerve fibers are totally crumpled and his situation of moving situation was not good. We have tested for all tests which I was established in this hospital, but there was no positivity. Then I asked her the history, what is your history? So she said, I am taking insulin twice in a day or something, very high dose. So that then doctor has concluded that this is because of these side effects, she is getting this movement disorder, not actually the any. So the diagnosis of neurological disorders is done perfectly by the use of molecular diagnostic. prenatal and postnatal diagnosis of genetical disorders. We can stop genetical disorders by proper counseling. Metabolic syndromes like diabetes or obesity has not done much work. I was thinking of doing a lot of work on this, but could not do further because of some restrictions. And then pharmacogenomics drug discovery, it is a great help of molecular diagnosis, especially next generation sequencing is giving great help in doing the pharmacogenesis work, especially in the drug discovery. Coming to my work, I have extensively worked in cancer research program at Jaslow Hospital. Some of them I want to put it here because it was really a breakthrough work in all over India as well as work. I have worked on circulating tumor cells, liquid biopsies. It's called liquid biopsies. Now you know that cancer has different stages. But when it goes to the metastasis stage, there is no cure for the cancer. And even doctors say there is no treatment. But because of this all innovations and the process, we can able to help the metastatic patient. That was my aim of an objective. There was a report comes that whether the cancer tumor which is inside, suppose lung tumor is inside, whether we can get some prediction from their blood samples, and that we did, and we found that the specific type of cells called circulating tumor cells, they are coming into the bloodstream and bloodstream, and those circulating cells can be mapped. Mapped means they can be profiled for genotyping by molecular methods, and we can study the genes, and then we can give the treatment to the patient, and that is working very well. That is also called the personalized therapy or targeted therapy. So this type of work has already I have done. I have achieved a lot of work in this. I have published several papers, and one of the papers by Praveen Jyoti Nauji, it is almost seen by more than 25,000 people. And I was also a guest editor for this journal, and this has been published in 2018. These are the cells which we have first shown that these type of cells are there. We have done the molecular profiling of also this. So this is a part of the work. I can give you the full idea about because there is no time. Next work which I am presently proposing is the immunotherapy of cancer. This idea I had a lot of years. I was thinking that whether boosting the immune response or making the immune cells, whether we can kill the cancer cells. Because what is the present situation is that there is no treatment. Treatment is radiotherapy and chemotherapy. These are the final alternative to the all clinicians. Whether there is any additional therapy which will not kill the cancer cells, normal cells. What is happening in chemotherapy and radiotherapy? The normal cells are getting killed, and that gives so much side effects that patient die because of this effect. So whether we can minimize this, and really I was fortunate, I was thinking of this, and in 2018 also, Dr. James Ellison from MD Anderson Cancer Center got a Nobel Prize in immunotherapy of cancer. I was very fortunate that I was working at MD Anderson at bioimmunotherapy department, and there only I got all idea of getting a targeted therapy. I have worked on targeted therapy in MD Anderson cancers. I am also very happy that I am a alumni of MD Anderson, from where the Dr. James Watson got a Nobel Prize. It was really wonderful thing. So in immunotherapy, there are major four parts are there. People can work on monoclonal antibody, which were obstructing the receptor. People can use the checkpoint inhibitor, whether they can obstruct the cycling pathways. They can develop the vaccine development. What was my focus? I am thinking of working on CAR T cell therapy, which is already I have submitted the project to the government of India. It has been approved, but at the last stage, the finance has not been provided, maybe because of the competition or less number of seats and this thing. But it was up to the final round. I have come to the CAR T cells and circulating tumor cells project was very appreciated by government of India, and I really pray from here in this national summit that they should encourage the people like us. We are not the influential people, or we don't have much support, but we can do better work in short duration because we know the technology very well. Now, I am also publishing, here also I have published one guest editor this year, 2020, on immunotherapy of cancer, future possible therapy for metastatic cancer. I am associated Journal of Cancer Metastatic as a editor as well as a board member. And I am fortunate that I am associated and they are really now on the PubMed. They have the Impact Factor journal also. So they have also again made me the chief guest editor for personalized medicine, where I have so many years I was working on targeted therapy of cancer. So yes, I will finish now. So this is my works. So coronavirus is also one of the area of work, but so far I have not started any work on this. So in concluding remark, I personally feel that the molecular medicine continue to expand rapidly, not only in the breadth of understanding science and technology used to study it, but also in the number of scientists and clinicians connect together to straighten their ideas to each other for betterment of human health and disease. I have shown the picture that there is a need of clinician and scientist collaboration is must in working in this area. Otherwise, you can't work. Therefore, I think clinicians should come forward to work with the scientists, and they can make a great breakthrough in years comes. I am thankful to you all for listening my talk. This is my number and email address. If anybody wants to contact me for discussion, they can definitely contact even after this meeting. Thank you very much. Bye.
Thank you so much, sir, for the very insightful talk, and it was great to listen a lot from you. So yeah, so now moving ahead, sir, can
Okay.
you close your presentation? Like
Yeah,
stop
yeah.
sharing.
Yeah, 1 minute.
Yeah, so we don't have as such any questions, I guess. So we'll move ahead. Okay. So yeah, so eventually if any question drops up, I'll ask you, sir. Okay.
Yeah, sure, sure. Anytime you can.
Yeah, okay. So yeah, so our next speaker for the day is Dr. Preeti Chunarkar ma'am, professor at Vidyapit Deemed University, Pune. So I welcome you, ma'am. Sir, can you stop sharing the screen? It's still on.
Achha.
You have to stop sharing the screen on Google Meet. Yes.
Stop presenting, no? Okay.
Yeah, done. Okay.
Okay.
Yeah, Preeti ma'am. Yeah, I guess ma'am is there. Ma'am, you can take over the session now.
Hello.
Yes, ma'am.
Yes.
Yeah, you're audible.
Yeah, thank you, Aditya. And
Yeah.
I'll thank the JAX Foundation for providing me the opportunity to deliver a talk.
Yes, ma'am. Go ahead. You can go ahead.
Yeah. Let me share my PPT with you.
Yeah.
Yeah, is it visible?
One. Yeah, it's loading. 1 second. Yes, it's visible now. You can just
Yeah.
put it in full screen. So
Yeah.
that would be.
Yeah. Now, is it visible for everyone?
Yeah, you can put it in full screen, ma'am. There's an option beside the percentage mark down. Down. Yeah. Yes.
Yes.
Fine. Now it's visible properly. Yeah.
Okay. Again, thank you, Aditya, and thank you, the JAX Foundation, for providing me the opportunity to deliver a talk. With respect to the all dignitaries and the students who's present over here, I would like to start my lecture, and I'm going to deliver my talk on the data analysis and the data centers, a need of the hour. Actually, my area of research is metagenomics and the bioinformatics, but as it is the summit 2020 of the biotechnology, so I thought to deliver the lecture on the current aspect of which the young generation must be needed right now. They must be serving and for the jobs. So as per the market research, the next 25 years are of the data analysis and the data. So I'm just delivering this talk for the students' purpose, because one of the motto of this particular summit is to create the responsible youth leaders in fostering business. And with respect to the all dignitary, I'll start my lecture. Now, all the biological data we will be able to classify into the three, like a sequence, structure, and the literature. So all the biological data you will be able to have only within these three domains. Okay, and we will be able to present that particular these three domain when we try to analyze the data of maybe a sequence, maybe a structure, or maybe a literature. There are the different types of tools or the different types of algorithms, techniques with the help of which we will be able to analyze the data. So the main purpose of the data analysis is always is that to answer the research questions, then to determine the trends, and to find a relationship among the different variables. Now, actually, what do you mean by then the actual data analysis? Because we are analyzing a data for the biological sector, and biological sector we will be having the three types of data. So, as giving you a definition for the data analysis, generally it is the clinic transforming and modeling the data to discover the useful information for business decision making. As much as good data you have, you will be able to filter that data, and you'll be able to present a very good data. As if I'll talk for the statistical methods, so as much as more samples you will be having to present the data, you will be getting such a good result. So as much as the sample number, so you'll be getting a good result. As much as the less sample number, then the result may vary when somebody else will present the data with the number of samples, and it will going to give you that how exactly we're going to make the decision so to go further with that particular study. So whenever we take any decision, just for example, in bioinformatics we used to say that we used to predict the future based on the exact present. We will not predict the future based on the past. But now for the data analysis, you need to take into account the past and future and the present data also. So whenever you need to take any decision in the day-to-day life, so what happened, you need to keep it in mind that what has happened in the last time and what will happen by choosing that particular decision. So means we will analyze here the past and the future. And exactly the same thing, the analyst, we used to say that them, the business analyst in the market, that what it means that they are analyzing the past data and they analyzing the present data so that they will be able to predict the future data. And that's what is called as a data analysis. Now there are the various, a huge amount of the tools right now available in the market to analyze the data. And actually, right now I'm only talking about the biological data. There are lots of different types of data which you need to analyze, and there are the different types of tools which are available to analyze that particular data. It's very, you must be very, everyone may be the familiar here with the R language and the R packages, then the Python, then the statistical analysis system, Java, MATLAB. These are the generalized tools with the help of which you will be able to analyze your data. Now, what exactly a data? So we need to categorize that particular data. Just for example, the data can be the quantities, means which we will be able to count it out, and maybe in the form of a quality, like the attributes, which we will not be able to count. But you need to give data exactly, just like I'll like to say here that what is the difference between the identity and the similarity. Exactly the same way you have the identity you will be able to count, and the similarity you will not be able to count. So the qualitative term and the quantitative terms, both we need to consider while processing the particular data. So just for example, I just have given here that if you have the sources of a data, you need to do, obviously for doing any type of a project, the student very first need to go through a very vast literature review. You need to find it out that what the people has already been done on that particular data, what are the different types of outcomes which has been shared through the published literature. So you need to take the surveys on it, and you need to collect the data. You need to keep it into the record formats. You need to have that what type of different experiments they have done, whether they will be successful, and there are the lots of it's not mandatory that every time whatever the research you are going to do, it will be fruitful. No, when we are saying that we are in the field of a science, obviously there will be the odd and evens. So we need to go with it. So you need to keep those things into the mind while doing a particular research or handling the particular project. So your data, you need to categorize that data into a different format. So it may be a comprehensive, it may be into the sample format. I just have given a very brief attribute over here in the form of a data, that how you need to categorize the data. And as per the data type, the categories will vary. If you are handling a biological samples, so data will be different. You are handling only a bioinformatics samples, in the sense you're just taking the samples from the data bank and then you are withdrawing some conclusion. So obviously at that time, again, this data quantity and the qualities will differ. Now, what are those types of the variables? Okay, so as I have discussed, that there is the qualitative variable and the quantitative variable. And qualitative variable, obviously, it is non-numerical, and this means they are categorical, and they may be a nominal or the ordinal. Now, what do you mean by nominal? Means just maybe a namesake data is very less to be considered, and the ordinals means maybe in the form of a series, in the form of an order. Okay, just why I have shared here some examples, that is the nationality, race, and the gender, and exactly same with the qualitative, quantitative data. So these are the, obviously it is the numerical data, and it may be a discrete or the continuous data. So discrete may be the things which you will be able to count, and the continuous data means with the things which you will be able to measure. So just for example, for a discrete data, the number of pregnancies. So 10 are the number of the pregnancies. So we represent a discrete data, and the continuous data means what is the duration of the Caesar. So that is not a time. So it will going to give you the continuous data. Okay, I shared here some cartoons, so that maybe a lecture will feel a little bit interesting. But there is again one message over here, that when you take a qualitative data and go, so there are the two types of a scientists which will deal with, okay, the qualitative data and the quantitative data. And obviously the both researchers are the best research, but still he'll start with the best and he'll end on the best. Just for example, now there are the various steps to analyze the data. As I have already told you, that the data analysis will always be based on what type of data you are considering. Here I'm just talking about the generalized aspects for the students, so that the various steps, like before data collection, what you need to keep in mind, that you are collecting a data. So what are the methods of the data analysis which you are going to implement? Then how we are going to process that particular data? You need to consult the statistician for analyzing that data in a very systematic manner, so that we will be able to come on the certain types of conclusions. And then you need to prepare a dummy table. So that is the before the data collection. And once the data collection has been done, so after the data collection, now you need to process that particular data with the help of the data processing tools, algorithms. Then you need to prepare the graphs and the tables. Now, till now, we have listened a lots of the lectures where you have seen the different types of graphs and the different types of tables, which is giving you the certain particular values. So based on these graphs and the table values, we will be able to analyze and interpret the certain findings. Okay, and then prepare for the editing and the presentation, in the sense that once you come on the particular conclusion, then you will be able to present it as a research paper. Now, as the two types of a data analysis are there, one is called as a descriptive data analysis and another is called as an inferential data analysis. So descriptive means you need to describe, there are the particular samples and you need to describe that there should be having the certain descriptions for that particular data analysis, those particular samples, and hence the conclusion must refer only to the sample. We are not talking about here for the whole and soul. No, we are only talking related with whatever the sample we have taken into consideration for dealing with that particular project over here. So in other words, this summarize the data and describe the sample characteristics. Now here we are again coming for the attributes of that particular sample, that when, what, how, all you need to give the answers to all these questions. Now, the descriptive statistics are obviously it is a numerical values obtained from the samples that gives the meaning to the data collected. And just for example, what are the descriptive analysis? So, mean, median, mode, standard deviations, range. It is the statistical measures for the descriptive analysis. And now, what are the inferential analysis? So, inferentials where you will go into have certain inference based on the certain test. Just for example, it may be based on the certain hypothesis. So use of the statistical test either to test to the significant relationship among the variables or to find the statistical support for the hypothesis. Now, guys, bioinformatics, it's used to say that it is an interdisciplinary field, but totally the based on the whatever the research and the experiment has already been considered and put into the format of the repositories, and from there we used to take the information or the data which we want to process further for the analysis. So there are the lots of the hypotheses, and based on that, like the people has already been worked on the experiment, and then they have generated data. Then based on the generated data, we are again making the certain hypothesis. We are preparing a different types of algorithm. We are doing a drug designing part over there, so that it will going to save your time, money, and the labor. So that is called the inferential statistic, that based on the something substantial. So the inferential statistics are numerical values that enable the researcher to draw the conclusion about the population based on the characteristics of the population sample. Now this is based on the laws of the probability. Now I don't want to go into the statistical part. Now the laws of the probability will going to give you the chances of the success, that how much percentage the particular data will going to capture, or what are you exactly able to say is that key. If you have this data and if you're going to process, so okay, there may be a chances of the 80% that you will be going to get a good result. And if the chances are less, just for example, if it is going to give you the 40 to 50% of the result, so just withdraw that particular algorithm, just filter the algorithm again and develop it once more, so that the rate of the accuracy will be more, so that you will be able to process that particular things. And obviously it is totally based on the level of the significance. Now within the statistics, I don't know how many of you are familiar, but within the statistics, as much as the significance of your data, that is going to tell you, okay, that is a good data, and you need to again go back and again recheck that particular you need to again go back and again recheck the things and again filter the data and come on with the good data. Just for example, here I mentioned the two tests, like the t-test and the ANOVA chi-square test, which is going to give you the inferential analysis. Now this is how we actually represent the data in generalized way, like a tabular format, so with the help of the tables. Then the textual format, like everybody is familiar with the book, magazine, scientific papers. Then the semi-tabular, where you will be having the literatures also and the data within the tabular format. And then the graphical formats, as I have already told that we have seen a lots of the graphs over here, which is going to give you the exact result, the analysis of the result, and it will going to take you to the certain conclusion of that particular project. So in such a way, you will be able to represent your data after analysis. Now, guys, directly move towards the big data, because the previously Poddar sir has given a very beautiful talk, and he has explained the simple sequencing methodology to the next generation sequence methodology. Like we were used to do the Sanger sequencing, then just due to the certain drawbacks, we move towards the automated sequencing. Then, or as automated sequences will not be able to handle the huge amount of a sample, then we move towards the next generation sequencing. Now we are utilizing the third generation of sequencing, but there is still a need of more. We are like scientific community always require more data to handle at a time. Okay, so that if you are having the huge amount of a data to handle, that huge amount of a data is usually called as a big data. So it is the current field which is booming right now. So what is the big data, how to deal with it? So obviously a big data will going to be deal with a huge voluminous amount of data, but the scientists and the research group are trying to do it in a very simpler way, that every person with the help of his simple PC and the laptops will be able to process that data through the big data analysis. Like you, if you have the very big data in front of you, you need to break down that particular big data into the small packets, and then analyze a small packet at a time with the help of your simple PC. That is a very simple basic concept behind it. And now there may be lots of the discrepancies also over there, that how to utilize it. There are lots of V's, that is the variables within the big data, and when and how utilizing that particular variables into the big data. depends on what type of data you are having. I have just given a normalized view about the big data over here, that if you have a biological big data, so you can do it with such by developing a databases, by utilizing the Hadoop. You need to have a very good hardware facility. You can utilize the machine learning algorithms or the deep learning algorithms over there. Okay, and this is what a data we are having, big data, and then we'll be able to store it into the cloud, and so it will serve our purpose to analyze the voluminous amount of a data, and that's why I move forward towards the big data. As I have already told you that what was a need of the big data analysis is that as eventually we are gathering the huge amount of a data, and in day-to-day life we are requiring the more and the more people to analyze the data, because the biologist or the biological people has generated a huge amount of a data, and it has been stored into the biological repository. Different types of a biological repositories are there. Now we have a data, and now we require good people, good personnel or the trained people to analyze that particular data. And as the current booming is the big data analysis, so a big data could be analyzed with the help of the big data tools over here. Okay, I have already been talked about lots of things, and for the current situation, as the complete world is right now facing a problem for the COVID-19, and huge amount of data is available right now, but still we are unable to find good vaccine over that particular COVID against the COVID-19, and we are facing the such a pandemic situation. So there where we require to analyze data completely throughout the world as per the age wise, as per the range wise, as per the different types of parameters, and then there may be a success into finding out the vaccine for the COVID-19. As I'm not being the expert into that particular field, I'll not comment on it, but it could be handled with the help of a big data to collect data throughout the world. Now, uh, my topic was a data analysis and the data centers. As I have told you, as the data is already available, and lot of data will going to be poured within that repository, biological repository. So now the people, we require a lot of a student who will going to do the data science or the data analysis, and from where as the repositories are there. So these are the few of the repositories which I would like to share with you, that there are the worldwide 10 best repositories of the data centers, which is collecting different types of data. There is one repository of the next generation sequencing also over there, which I have shared, from where the student will be able to take a data for doing a particular type of a bioinformatics research. And these are the very familiar one, biological data centers, that you are having a lot of biological data centers, the biological repositories, where you'll be going to get a nucleotide data, protein data, structure data, literature data, okay, comparative genomics data, and from there you will be able to take the data and analyze the data with the help of a data analysis tools. So guys, I think I have taken enough time. Thank you. And if there are any questions, I'd like to take. Hello? Hello?
Yes, ma'am. Yeah.
Yeah,
Thank you
yeah.
so much, ma'am. The
Yeah.
session was really great. And we have one question from Hariom Gupta saying,
Yes.
what are the importances of hypothetical and traditional data, and how far you may consider it from the experimental data?
See, we've come on the one particular hypothesis based on the experimental data. That's what I said, that though the bioinformatics is interdisciplinary and maybe the mostly based on the hypothesis, but those hypotheses are coming from the experiment, which the people has already been done, and they have deposited their data into the repository. So we are making the conclusion based on those experimental data. So we are not just making the conclusion just as a way. We are making it based on some substantial data, so that as per that available data, then you are making the hypothesis. So actually it's good.
Okay, ma'am. Yeah, thank you so much. Yeah, that was the only question. Okay.
Okay, thank you.
Yeah. Yeah. Okay, ma'am. Thank you. Yeah. So now moving ahead, our next speaker for the day is Dr. Swarup K. Chakrabarti, co-founder and director CEO of STEM Bionics Private Limited, Kolkata. I welcome you, sir.
Yeah, thank you, Aditya. So should I go ahead and present now,
Yes,
right?
yes, you can go ahead now. You can present the screen. Yeah.
Okay, so, key too, right?
So there is a present now option there in the Google Meet down
Okay,
below.
okay. So,
Yeah.
Yeah, entire screen.
Yeah, entire screen, and then just switch to the PPT that you have opened.
Oh,
That
okay,
should
okay. So I have
that should do it.
okay.
Yeah.
Okay, now share.
Yes, sir. The PPT is visible. Just can you make it full screen? Then that would be best.
Yeah, I made it full
Yes,
screen.
done.
So you can hear me well, right? Okay, sure.
Yes,
Go
yes,
ahead.
we
Okay.
can hear you well.
Okay.
You can go ahead. Yeah.
Okay, so thanks for all your patience. I know we all have been listening for such brilliant talks, and it's a brilliant symposium. And I'd first like to thank Jack's Foundation for giving me an opportunity to present some of our work in a prestigious national meeting. So the title of my talk is Cell Therapy in the Precision Management of Human Disease. So if you look at it, I keep it a little bit broad. So with a limited time, I don't want to cover a lot of other diseases also, but I'm going to cover one of the diseases which we have been working, which we have been using cell therapy to treat some of the disease area is called the infertility. So I will show some of the data. And throughout my talk, I usually keep my entire talk very simple and make it very simple, and always my message to the students who are studying biotechnology, who want to be a scientist, always try. I keep telling everybody, try to keep your presentation very simple. Forget about the papers. You have to use a lot of scientific jargons, but for the presentation, because see, there is a reason, because whatever the work we do comes from the grant money, right? And these are all comes from the taxpayer money. So ideally, you need to learn actually how to communicate your science, to the lay people. So this gives us an opportunity to present the talk in even a very scientific, complex thought in a very simple manner. And the better you are at it, I think you'll be good at it for the things. So this is our talk, and so talking about the cell therapy, start with some of the drugs. So these are we all aware of these drugs. These are the drugs we know. One is synthetic drugs. It comes in a prescription form, so you get it from the doctors, or you can go to the over-the-counter and get a pill or capsule and take it and cure your disease. So the issue with these synthetic drugs actually, so it takes a long time, to synthesize and go through the development process. And I think earlier speaker has already told about, the challenge of, getting a drug into a market. So it amounts of money and going through all the tedious process of clinical trial, phase 1, phase 2, phase 3, and even the post-marketing marketing authorization. So it's a tedious process. And if you come out with 10 different targets, chances that only one target you can bring it to the market. So it involves a lot of time share, a lot of cost and everything. So even if the money is very short or limited, it is very difficult to go this kind of thing. Another option is already one of the speaker has described is the repurposing of the drugs, and that limits the amount of time and the cost also. And there is another drug called the natural drugs. So it's no different from the synthetic drugs, but there is a difference is that because you don't have to some of the compounds, we already identified in some of the natural drugs, and we have our agency like Ayush, they are doing a fantastic job, in popularizing these natural drugs. So these compounds are already there in the plant. So you don't have to make or synthesize it. All you have to do, just extract the compound and and take it. So this is a good stuff. So these are the two drugs. They are in the standard care of practice. You go to the doctor, they know about these two things, pharma drugs and also the natural drugs. But there are on another type of work, and that's actually trying to that will revolutionize. I believe personally it will revolutionize the entire medical science. Is called the living drugs. So the name is kind of explanatory. What is the living drug? So it's the biologics. Basically, it's the biological drugs. These are the two examples of the stem cells and also the immune cell. But there could be some vaccine also. But just to make it simple, so I'm going to talk a little bit more about the stem cells. So the stem cell, we all have stem cells in our body, right? And the stem cell has a regenerative property. So basically, if you have some in your damage in your body, so we have the stem cells in our body. So somehow they can identify the damage, and they can go to the damage site, and it can heal your damage. So this is fascinating. It's a miracle drug. But another thing is that if you don't have a proper functional stem cell, or you have the low number of stem cells, we can isolate the stem cells from your body, and we can just expand it in a large quantity in the laboratory using all the guidelines by the Indian Council of Medical Research, or DCGI. So there are the guidelines out there, and it is a very clear guidelines what you can do. You can't do an extensive manipulation. You can do a little minimal manipulation. So I'm not going into all the regulatory details. They are in the public domain. You can go to visit the ICMR sites, and you can find all this regulation about this using of the cellular drugs. So stem cells. So basically, so as I said, that if you don't have a limited number of stem cells, we can take it out of your stem cells, we can grow it in a larger quantity, and you can give it back to you. So this is one approach. This is a transplantation is called autologous. So your cell going into your cell in your body. So this is an autologous stem cell transplantation. So there is another one is called this allogenic. So probably it happens is that you may not have a good stem cell, so there is no point of isolating your cells, right? So in that case, we might be thinking that you might be going for a donor. So and you get the stem cell from the donor, and then you can transfer it back to you. And the good thing about the stem cells, about specifically the one of the stem cells, they're all the mesenchymal stem cells, because there is always a chance of transplant rejection when you are, so the stem cells does not necessarily require that kind of the matching. This HLA and matching because of this low immunogenicity of the stem cell. So if it is an allogenic cells, which is often called off the shelf, you should be able to take it, and you should be happy to take it. So good thing about this allogenic gel. So I'm not proponent of this allogenic therapy, because I do the just the reverse. I do the allogenic. I do the autologous transplantation. But the good thing about the allogenic stuff is that suppose a patient come to the ER, right? The ER means the emergency. So in that case, you badly need to have a stem cell supply. So you don't have the time, right, to isolate your stem cells from your body and get it to the lab, expand it, and give it back to you. In that case, it will be very nice to have a stem cells ready from some other sources, so that doctors at the ER can immediately go for a stem cell transplantation. So the off-the-shelf therapy, off-the-shelf treatment like allogenic stem cells, and they are fascinating. And I think there are a couple of companies in India also working in collaboration with a couple of government organizations also. They're trying to bring this all these the one size fits all, though it's just violates the principle of precision therapy, but sometimes it's fascinating. So, but I'm not going to explain to that on details, but just to give you an idea about the two types of stem cells and what would be the approach. And there is another cells called the immune cells. I think one of the speaker already explained it. It's actually already explained that suppose if you have a tumor, somebody, I guess we don't have it. Some patients have a tumor, but somehow there is we have this defense mechanism in our body. So the tumor cells are supposed to be killed by the T lymphocytes or the NK cells. They are in the body. That's their job. But somehow, what happened in the cancer cells, when you have a cancer, they recognizes the ability of the tumor cells to look at the foreign. So they don't exert their immune response or whatever the back response to the tumor. So in that case, what you will be doing actually, you take the cells, there could be two options. One is that there could be two cases. The one case is that you don't have a sufficient number of NK cells or T cells, and there could be another case is that you do have the number of NK cells but T cells, but somehow they are not functional. So how do you do that? So the two options is that if you don't have enough number of the T cells or NK cells, it will be an easy fix. You take some of the cells from your blood and you isolate it and you just grow it in the cells. You can culture it in vitro and make more numbers and just give it back to the patients. But what if the function is missing? So in that case, the revolution. This year they got it. Or whatever. So you can introduce by genetic engineering. You can introduce some receptor at the T cells or maybe in the NK cells, and then you just infuse the engineered cells into the cancer patient, so that the receptor will identify the tumor antigen on the surface. So once it gets identified, then the other actions will be activated, and eventually they can lyse the tumor cells. So it's a fascinating area, but the one thing is that it is not in the mainstreams, because because for my business I have to go to the doctors and I have to teach them, because we cannot prescribe any therapy, right? So we have to convince the clinicians to, and the clinician will convince the patients to take help of the cellular therapy. So I meet every day with a lot of doctors actually. So we have to convince them that though they are kind of skeptic about taking these living drugs as the standard care of practice, because but I'll tell you, I bet you any money that within a year, within a decade, I think this is one of this will be integrated into the standard care of practice. What are the stem cells? Those who are new to the field, I already mentioned that regardless of the source, they are capable of dividing and renewing themselves for a long period of time. They are unspecialized and they give rise to specialized cell type by differentiation. So this cartoon is showing that you have a stem cell, so they can replicate, they make more of the stem cells. At the same time, they can do the differentiation. So they're they can go to any kind of cells. It could be the neuron, it could be the endothelial cell, or it could be any cell types. So there are two properties. One is the self-renewal, and another is the differentiation. So this is very simple way of describing a stem cells. So now the question is that, for therapy or any other application, so you need to know the source of the stem cells, right? So these are the type of stem cells you can get it. So there are three different types. One is the tissue stem cells. So these are all endogenous. And so all you have to do, tissue, it's in the muscle. You can get it from the bone marrow, you can get it from the nervous system, and get it from the skin. So, and another is that, and these embryonic stem cells. So these cells are created from the inner mass of the blastocyst. So those who don't know, there's a blastocyst. So when the embryo and the ovum and sperm fertilize, they give rise to an embryo, and then the embryo forms from the embryo we born. So you have this inner cell mass in the blastocysts. So that inner cell mass can be collected from the blastocyst, and you can grow them out in the laboratory and make it an embryonic stem cell, or you can make it a line. So I'm going to show you some of the exciting data, which we did in collaboration with one of our participating institute or one of the collaborating institutes, and I will explain in a minute. So that was a grant. That was the first grant in this country, and that was funded by Public Agencies Council of Scientific and Industrial Research back in 2018. We took a long time. It took us almost more than a decade to publish this paper because of the many stringent regulation and everything. But eventually, we got it published. So I'll tell you in a minute how to make embryonic stem cells from the blastocysts in a couple of slides. And there is another one, the iPS. See, the limitation with this tissue stem cell and embryonic stem cell is that it's kind of invasive. Forget about the skin. Skin may not be so invasive, but other like bone marrow and muscles and invasive nature. And also you don't get a tons of these stem cells from there. And blastocysts, it's a lot of people think this is kind of, I'm not going to the trouble of, what is ethical and non-ethical. But the thing is that the embryo destruction, for research even also, it's sometimes illegitimate for many of the Western countries. But But the time we started this journey, so in 2008, I joined late. I was not there. I was in the US at the time. But my participating institute, they got a lot of bunch of discarded embryos, and they are not going to use the patients are not going to use it anyway, right? So you can use it, and at that time the regulation was there. Actually, you can use in any discarded embryo which is not going to be used for any application or anything. You can use it for research. So that's why the institute got the CSIR funding, and I'll show you as I promised you. And there is another one is the IPS. So there is some issue of destruction, and this is the problem of the numbers. And so based on this, considering all this limitation, back in I think in 2006 or 2007 in Japan, they developed this induced pluripotent stem cells. So this is that I forgot the name of the Japanese scientist, got a Nobel Prize for that. So so basically it's a very simple procedure, but it's an outstanding concept. So you take your skin cells. So skins are very easily accessible, right? So you take your skin cells, and then they are differentiated cells, which means they have a definite function. So you introduce some of the genes which actually make stem cell as stem cells. So we know what are some of the genes, like CMYC and from some of the proto-oncogenes and OCT4 and all these things. There are 4, 5 genes. So you can introduce the genes by or knocking the genes into your skin cells, and they are going to be expressed in your skin cells. And that particular gene, actually, when they get expressed, they let the differentiated cells to de-differentiate, and eventually go back to it can convert it to the embryonic stem cells. Or I shouldn't say the embryonic stem cell, it's the embryonic-like stem cell. And as soon as you get the embryonic stem cell, now you are free to make, in culture, because this condition, you can manipulate it, in vitro. So you can make them to neuron, you can make them in vitro, or endothelial cell, you can make sperm, or not sperm. I'm not sharing some somatic cell type, but there are options out there, and there are culture media out there, and a lot of people, a lot of scientists are working on, how to develop a differentiation protocol in vitro to make them accurately and make them functional. So this is also an IPS cells. So as I told you that, we got a fund from CSIR to make this embryonic stem cell. This picture is showing that we can make that embryonic stem cell from inner blastocytes. And the project because it was not the aim of the project to make an embryonic stem cell. The project was aimed to how to use the embryonic stem cell for therapeutics. So we thought of, once we get the embryonic stem cells, so our plan was to, convert them into hematopoietic stem cells, because right now, as far as the ICMR guidelines, these are the most well explored area of blood-related. So you make the hematopoietic stem cells, and you can treat a lot of blood-related diseases like anemia or leukemia or sickle cell anemia. So that the initial plan is to make the hematopoietic stem cell. So this picture is showing that actually you can not only can make the embryonic line from the inner cell mass, from the blastocyst, from a discarded embryo. I'm telling you again and again. So this is not a viable embryo, it is discarded, and you can make the embryonic hematopoietic stem cells. So how do you know these are hematopoietic stem cells? Well, phenotype says, but also these cells are extensively characterized through CCMB, the Center for Cellular and Molecular Biology, and also other things. So we did a FACS analysis, because the hematopoietic cells are they have a marker called cluster of differentiation marker, CD34. So they are characterized by FACS. So they have a marker, so you can know that is the marker. And not only that, we have the expertise to do the genotyping. So we did the additional genotyping also, just to make sure the cells you are working with actually they are hematopoietic stem cells. But how do you know they are functional? So one way to know that these cells, these cells can be cultured in vitro. So think about the situation. So you make some in a petri dish, in a small petri dish, you make some hematopoietic stem cell, right? But that stem cell that hematopoietic is not enough for transplantation. So you need tons of hematopoietic stem cell for transplantation. So your approach will be to whether you can actually propagate. You can multiply the stem cells in the culture, and you can still retain their characteristics, all these things. So we extensively characterize that, and we can make more of these hematopoietic stem cells. And we didn't go too far beyond of that, because for other issues. but actually these are the, actually you can make, functional and actually a transplantable grade hematopoietic stem cell for some other, to treat some other blood related diseases. So we published the study last year in Journal of Stem Cells. It's a leading publishing agency from the US. And so these are some of these, some of the uses of the potential use of the stem cells. See the point, the potential use of stem cells. Don't get intimidated that stem cell is going to cure all of this, but there are promises. There have been a lot of clinical trials going on, and stroke, traumatic brain injury, bone marrow, spinal cord injury, muscular dystrophy, diabetes, and everything. But it's not yet clear. It's not yet clear, but it's a lot of scientists in the all of the world, they are working on it to understand the safety and efficacy of the stem cell in the treatment of various diseases. So just to give you an idea that it's a miracle drug, so it can treat many other diseases. So as I already told you that this we have a responsibility, social responsibility for the taxpayers, and these taxpayers they buy all these books like magazine like Time and all these things. So these people and this magazine people they started highlighting all this achievement of stem cells into one of this issue. Like see the Time stem cells and stem cells music, and there is another book also which we all know that Dr. Siddharth Mukherjee has published a book. So it's I think a long time back. Actually, so basically this is an awareness, and the people are very excited about that. Maybe with they are. I think the scientists are more respectful now because they know that they are the scientists back in the they are not only the doctors. The scientists actually they are working behind the scene, and they are constantly working on to bring out the therapy for the patients. So this is good. This is a good thing. So as I said that I'm going to focus mainly on infertility, but we all know about IVF. I don't think so I have to explain that. So what happened is that most of the women either they are working or for some reason they tend to start their family late, maybe in the 30s or 35. So the problem is that the thing that they have a problem in achieving conceiving in a very natural way. So we often see that most of the women at above 30s or something, they have to take help of the in vitro fertilization to have a baby. So this is the y-axis is showing the live birth per cycle. So per cycle means make it simple, it's per attempt. Okay, so look at this figure. So this is a figure from I think from Center for Reproductive Medicine or some of the, I forget it. It's from 2003 to 2009 data. They look at this, the success rate of, though the IVF was discovered almost 40 years, 4 decades back. Look at this success rate is almost 40%. But if you go to the higher age patients like 35, the IVF success rate is going down, and above 40, or 42, it's almost 5%. So a technology with the success rate is a 5%. It's hard to believe is actually a technology, right? But and then this is another issue. I'm just talking. I'm thinking about the patients. See how the patients have to they have to go to the travel to get a baby through IVF. Look at the cost. Per attempt, the cost is average is about 2 lakhs, right? So I will show some data where we use some of the patient. We have the patients who has actually multiple failures, 3 or 4 failures. So if you think about the 3 failures and you consider the amount of cost, it could be 8 to 10 lakhs. And not only the cost, the most important is the time share. Sometimes it may take a decade and the suffering and to have a baby. But they do it for emotional reason. So we considered, I think we need to do something. This technology is a great technology. We're not going to change the technology, but we need to come out with certain innovative idea to improve the success rate of the IVF. So so this is our hypothesis. But first you need, but I'm telling you that what is your embryo. So if you look at this, the cycle. So every month, it's after menses, the endometrium or what it shades, and you again get your uterine lining, the proliferative and secretive and post-menstrual phase. So your embryo implants between day 20 and day 23, known as the window of implantation, when the endometrium receptivity is the maximum. So lack of which results recurring implantation failure. So if you have a frozen, you have an egg or embryo. So the doctors usually they wait for 22 and 20 days, that's window implantation, and then they transfer that, the frozen embryo in the particular, day, 20 to 23, right? And so, so we thought that maybe this is the reason where, because this is your endometrium has to be very receptive to accept the embryo, embrace the embryo, and let them grow. So I think something is missing over here. So we thought that using cells of your own body when injected to your uterus can increase the endometrium receptivity during window implantation and augment your chance of conception. So this is, see, this is there is hardly one paper published in Japan. So we don't know. This is just, we just came out with the hypothesis. So we thought that let's isolate the cells from. I'll tell you what kind of cells this is. Isolate the cells from the patients and give it in the proliferative phase. And it may have the cells, the stem cells, because they have the regenerative property, they can help to grow, not only grow the endometrium, but they can also augment the receptivity. So that, once you have a good, ready receptive endometrium, then the doctor should be able to transfer the embryo and you will have a baby. So that was the hypothesis. We didn't know anything about 4 years back that it is going to work or not. So that's the entrepreneurs will do. Then they just took a chance. So sometimes it works, sometimes it doesn't work. But in our case, it worked. So so this is an anatomical location of the endometrium stem cells. So they are there. So it's not that we are putting something foreign to the endometrium, right? So you have these two layers of cells called functionalis and a basalis. So look at this basalis first. There is some progenitor cell and stem cells. So they are there in the endometrium, but somehow they don't know how to divide or or maybe they are quiescent. They don't know how to divide it actually. So they are waiting to have a trigger to amplify or proliferate and make it a receptive endometrium. So as I said that, considering the stringency of all this cellular therapy, so we have to register our trial in called ISR and CTR registry. There are 4 registered agencies around the world. So this is an UK-based registry. So we have our SOP, the standard operating practice. So we got a review and we had to fix something and then got it approved. And finally, we are able to actually publish the work. So this is our the title is that the effect of administration of white blood cells into the uterus on pregnancy outcome in infertile patients using in vitro fertilization. So our approach was very straightforward. So we usually take 10 ml of blood from the patients who had at least 3 failures of recurring miscarriage or 3 attempt of failures, right? So just 10 ml of the blood, and we isolate this, the white blood cells, WBC. It may have some stem cells also, but predominantly white blood cells. And then we give the cells to the doctors because we don't have that kind of license to do it. So the doctors, the clinicians, they infuse that, precisely. That's why I tell the precise management. They infuse that, WBC cells precisely into the uterine cavity under a ultrasound guidance. And the first, our customer or client, we have collaborated, fortunately enough, we collaborated with Dr. Buddhanath Chakraborty, and you may not, he was the father of our IVF, and actually he was almost the chair of the ICMR board in the art sections. So we collaborated with him actually, and he has been doing all these things, just we give him the therapy and just putting this into the patients and patients. So this is just a trial result. So now I'm giving the summary. So look at this summary. So this is some of the demographics. So this is basically a clinical study. So I'm not going to go into all the complications of BMI, everything. But one thing I like to restrict your attention is the success rate. Look at the success rate is 56.35%, and look at the average age is 36, right? So see, and if you remember the figure I showed you, so it's a huge increment. It's a huge increment of your success rate from about 20 to 25% to 56%. So it's a 55. Actually reported 96 cases, but we have done more than 200 cases. But so it's a huge increment. And remember that these patients has already had 3 failures. So if you consider failures, success rate could be more than 56%. But there are some miscarriage, of course, there. So so we are very excited. So we have been continuing this study with Dr. B.N. Chakraborty, and then And the type of infertility the patients we got, there are a lot of unexplained and the recurring miscarriage. PCOS is polycystic ovarian syndrome. And the success rate varies. Some of the 61%, 57%, and sometimes we don't get any success rate also. And of course, there are some miscarriage also, because it cannot be foolproof. A technology cannot be foolproof. Even you use the cell therapy. So we got some miscarriage also, but that's okay. That's part of life. So we cannot make every patient happy. So, but we actually, but the good thing is that we tried, but somehow it didn't work. But actually, so we can augment the pregnancy rate about. For unexplained one, it's fascinating, 61.6%. And even for the recurring miscarriages, the 29 cases is not 3 or 4 cases, 29 cases. And look at the pregnancy rate, 55.14%. So that's a huge increment. And then we try to understand the mechanism, because see, that is one of the good things and bad things about clinical studies. Actually, it's very difficult. You can always design your endpoints, but you don't know what's going on in the body, right? So just like in animal study, you can actually figure it out, the exact mechanism, what is going on. In human, you cannot. It's unethical. It's unethical to go and to see and what's going on. So, but there are a couple of parameters we can measure, which is very non-invasive. We look at this endometrium thickness and vascularity, and this can be done by ultrasonography. So we did look at the pre-treatment before cell therapy. The endometrium thickness is 8. After post-treatment, there is an increase of the endometrium thickness, and it's statistically significant, and also the vascularity, right? So basically, the take-home message is that cell therapy is actually making this endometrium more responsive in terms of their endometrium thickness and vascularity, and these are the key parameters in assessing whether it's a good endometrium or whether it's a bad endometrium. And look at the non-pregnant group. You see the endometrium thickness, though there is a trend of increase of endometriality, but not statistically significant. And vascularity, of course, there is a little bit, but it's not statistically significant. So our conclusion is that maybe the increase of endometrium thickness and the vascularity is actually improving the success rate of in vitro fertilization. So this is, as we said, this is what we think is happening. So this is our, look at this picture. These are peripheral blood mononuclear cells, or WBC. They can, once you put it in the proliferative phase, infuse it, they can differentiate and make more endometrium tissue. And they can also differentiate into endothelial cells, so that you get a highly vascularized endometrium, right? And not only that, I didn't mention it's not the cell-cell contact alone. All this cell therapy, they have a bunch of growth factors, so they can be released, including exosomes and other things. So they can get released from these cells, and these these factors can hone in to that endogenous stem cells or endogenous stem cell, and they can give that instruction to proliferate and differentiate. And not only that, as I said, that in the window of implantation, this is the crosstalk between a blastocyst and endometrium is very important for an effective implantation. So what this growth factor does, actually, there are a lot of reports out there, so it can change the Th1 to Th2. So just make it simple. Th1 is actually produces pro-inflammatory phenotype. Th2 is the anti-inflammatory, and Th2 is anti-inflammatory. So Th2 as an anti-inflammatory. So when this switch, it turns on this switch, and what it does, actually, it activates the eNOS, endothelial nitric oxide synthase. So once it goes off, then it can make more of this endometrium tissue. So this is what we think is happening based on the literature reports and everything. We cannot do all this invasive study in what is going on in the patients. Is unethical, but we can come out with this model. Actually, this is those things. This is happening inside in the patient's body. So this is finally, I'm going to conclude. So you carry your own repairing kits in the body, but somehow you don't know how to use it. But we can give you, by instruction, how to use it and how to trigger it and how to heal yourself heal yourself in the body. So like to acknowledge, thanks and acknowledge our collaborators, IRM, which has become the ICMR Institute, CCMB, and then BHU, IIT, Genome Foundation, and we got the funding from CSIR and also from some intramural funding from IRM, ICMR. So thank you for listening, and I'm going to end here, and would love to answer some of your questions if
Yes,
you have.
sir. Thank you so much, sir, for the great talk. Now we have some questions here. Praveen sir is asking, did your company have ICMR approval to do the cell therapy?
Yeah, so we have this. See, the thing is that we have the IESC, Institutional Ethics Committee, right? So we have this IESC, and we have this, this ICNR and SAR approval from the body, right? And that's an international body, right? So it's got, and that is approved by the WHO. So directly, we work in ICMR Institute, but we don't have the, well, yeah, what you were asking actually, but we have all these legal documents. So yeah, so indirectly it's either yes or no.
Okay. So Hari Om Gupta is asking, what are the roles of stem cell in the weakness of eyesight? Please explain as an example, if you can.
Yeah, eyesight, right? So there are a lot of uh, the one of the eyesights I know, it's an advanced AMD, it's age-related macular degeneration, right? So that is one of the a lot of people, older people, they have this, their retina, get somehow it's a natural progression of the as you age. My mom has the same thing, like an acute advanced macular degeneration. So I think there are a couple of reports out there. They implanted some stem cells, and they claim that they cure actually this the progression of this age-related macular degeneration. And of course, and there are some reports out there also, some retinal injury, some cornea repair also. There are some reports out there also, but they are mostly one or two case studies. But I don't know whether there is any comprehensive study on this.
Okay, sir. Thank you so
Aditya,
much. Yeah.
Aditya, just I want to ask one question to Dr.
Yes,
Sri.
yes. Go ahead, sir.
See, you got a ICMR approval. You got the stem cell committee, correct? Institution stem cell committee. And did you get ICRT approval for this project?
I see. Yeah, ICRT. We haven't done it actually. So, but we have this one. They are working on it, and since this institute has become an ICMR institute, and I personally talked to some of the ICMR people. They came to and looked at things. Yeah,
that is because
yeah.
I am also
So I think
with
So
this problem.
that's one of the use the Apex committee and IESC. Yeah, that has to be.
You
But
should have the ICSRT committee's approval.
Yeah, and think about
stem cell therapy.
Yeah, and also we don't call it a stem cell therapy. This is, I showed you that this is all WBC. There hardly any stem cells are there. Maybe there is very minuscule amount.
That is the thing. They are all people are doing as a stem cell therapy. Everywhere I see, and that is totally wrong. If you call as a stem cell therapy, you have to really differentiate those cells into the similar type of cells. Because I work on stem cell therapy, I have been work. Even I had the institutional committee at Just Look, but then further that ICRT committee was not formed that time, 2006.
Right.
But I think that approval is required to do any type of cell therapy to the other patient, because doctor comes and asks us, okay, give us a cells. Many doctors in Just Look hospital, they used to come to me, sir, आप cells grow करो, मेरे को दे दो. So
yeah, I know.
That's unethical actually. What I feel.
Yeah, but still, this is an early phase of these things, and doctors are sometimes very skeptical about this. But we have a couple of documents also there, because we have this.
What documents?
I mean, who approved the protocol approval?
From where?
I showed you some data. ISN theater, this clinical trial registry.
Which is that actually? No, because I still remember, because one of the project on COVID has been refused because there was no committee has been. They say that it will take about 6 months to get your approval or not approval. Otherwise that was a very good standing project, which has internationally it was approved actually. But I could not go further because of not having this approval, and they have not ready to even say yes, okay, you go ahead, make a institutional committee as a committee, and then I know the procedure. But we stuck up with the ICSRT committee, which is ICMR EPICS committee. So that's why I had a question. And this type of work, many people are forwarding actually, doing it as a stem cell therapy. which I just want to say this is not a stem cell therapy. That is only my
No, it's not. It's not just cellular therapy. Just
cell therapy, but you cannot charge the patient. No, very heavily people are.
no. We don't charge. We don't because it's an still.
So
You know,
you
as
do
long
free
as
of
you're
charge.
as long as you're in an experimental phase, you know, you should not charge
Yeah.
it. Yeah.
Thank you very much.
Yeah.
Yeah, thank you so much, sir, for the valuable input. So yes,
Let me
so I
see.
guess, yeah. So now we'll move ahead. So with the last speaker for the day, Mr. Radur Samadar, sir, UG Research Assistant, Kolkata. So I welcome you, sir, now.
Yeah, hello. Welcome everybody. Thank you for inviting me. I thank Jack's Foundation and especially Deepa Patil. So, unlike all the eminent speakers here, I'm just an UG student of biotechnology. I'm in Amity University, Kolkata, and I have been called here because I am a COVID-19 researcher at the lab and also a frontline biotechnologist at the Fearless Hospital. So if you say, I may start with the presentation.
Yes, sir, you can start with the presentation. You can share your screen now.
Right. Yeah, so is it visible?
Yes, it's loading. One second. Yes, it's visible now. Just make it full screen. Yeah.
Okay.
Yeah,
So,
you can go ahead.
yeah. So welcome everybody. This is Rodhu Samadar. I'm from Kolkata, and as you see, I'm working with Fearless, and also I'm a WHO registered COVID-19 volunteer for the pandemic response and preparedness. So I will talk about nanotechnology today a bit, and also SARS-CoV-2. Also, I am a active COVID-19 researcher with Fearless Hospital only. And on that note, we have sort of collaborated with many companies, many private institutions, and also government. We are trying to get a government funding on some projects that we have formulated. So I'll be speaking on that. So speaking of the topic, biotech at this era, we know everything about it. Coronavirus has struck and COVID-19 pandemic is ongoing. We are amidst the search for vaccine trials. All the companies are they are struggling day and night to find a cure for this virus, to at least to stop the spread, if not cure. So we are dramatically being affected in many aspects of the societal aspects, not only health but our economy is on a downfall, our social mobility and all that. Even I'll say our physical and also our mental health. Everything is being affected at once. So biotechnology research, improvisation, intelligence, goodwill, generosity, and I believe the integrity of the entire scientific community at once is what we need, and it is the need of the hour. So I'd like to thank our government that we are so powerfully we are struggling at so many research attempts. So the economic drive is also fragile at this point of time. But talking of biotechnology, this era is, I would say, a boon in disguise for biotechnology, because we are receiving a huge number of research fundings. Our research, the attempts have it has risen to a huge level, and within this 6 months, the number of papers coming in, every aspect of the research and its integrity has rise. So I would also like to say that the funding and also the venture fundings that are going in to the research of the medical research, especially clinical research, I'll say at this point of time, is it's booming, and we must take the good opportunity and we must use it to our scientific progress. Research always gives way. We all know that. So there are few researchers, like we know there's the RT-PCR. Everybody talked about it. Our eminent speakers have already mentioned what all they are. So research, we are still researching. We are coming to the point. We are trying to come up with better ideas which are economical, a solution which will be faster. It will give us, it will save a lot of time. It will be economically it will be more helpful for us. And as we are a developing country, we will always strive to get things done in a simpler, in a very less, less expenditure, where less expenditure is involved. So here are some results that I have cited, that we have a COVID-19 swab testing that's going on, and that is cheaper, and it just takes 10 minutes for just the the RNA extraction, and after that you get the result. We have the antibody testing that like helps you to mark whether you have been previously affected with the antigen or not. And recently, we in the news there was the Feluda, which was in collaboration with CSIR and Tata. They had this CRISPR-Cas9 based. Basically, this was a gene knocking based coronavirus detection tool, and like this also. With technology, I'd like to say that this Arogya Setu app, this was recently uploaded by WHO for standing out in like improving the rate of community spreads, and exactly because we are so dense in case of societal parameters, and our so like demography is such in a way that we need to be aware about these things. So as Preeti ma'am mentioned in the earlier talk, that data is the new goal. Yes, so it is. At this point of time where we are, we will have to strive to bank on data. We will have to strive to bank on in silico studies more than in vitro. And at this point of time, we know that the labs, the working professionals, the entire structure, even the logistics of the research and development, everything is facing, it's kind of at a standstill. If not at a standstill, it's somewhat affected, like every other aspects of the society. So we should be aware about it, and we should drive our courses to ways where we can go with scientific predictions with the help of in silico studies. There are huge data banks of protein structures and whatnot. We have the genomics and proteomics. All the data are available. So we should be trying to save our resources in that way, and try to motivate and motorize our way through that road. And also, I would like to mention about this COVID-19 vaccine that is under clinical trial among the other 12 and or the 15 that's going on. The multi-epitope vaccine, which will be they have performed all the molecular docking and every studies that has been performed for that vaccine on an in silico based environment, and they are only going to perform the clinical trials on real patients, and they are only going to formulate the drug, and that must have taken 3 to 4 months in the lab, and they were applauded hugely for this, for this management of resources in the smart way. So yeah, I have cited the example here. You can check it out if you're interested. So where does nanotechnology come? So nanotechnology has a very broad aspect at this point of time, but nanotechnology and biotech, when these two comes together, as simply as the term suggests, that we study the materials and divide it at a nanoscale, less than 100 nanometers, and that is what nanotechnology is. But in COVID situation, how can nanotechnology help? Now, nanotechnology not only promises to devise new systems and their more efficient usage and their development, but it also gives a broader spectrum for research and development. So with the help of slight modifications to what already we have got, we can develop a much cheaper product, and for it can be used widely. So of course, taking into the consideration that we'll have to go by all the stringent policies for the formulation and publicizing, but taking that into consideration, this is what nanotech offers. So here I'd like to explain some of the aspects that we have been working on. At our lab in Fearless, we are trying to work on basically a PP kit that we wear. So we have taken that as a control, the original PPs, and the waste disposal of these PPs are really at a problematic state at this point of time, because you can imagine, from one hospital we are generating 2 trucks of biohazard waste. So at this point of time, it's almost 4 folds to 5 folds. So we are trying to optimize how a self disinfection can be done on a PP kit with the help of intercalating silica gel formats, and we are trying to develop something like that. So We have also approached to the ethical committee and we have got the ethical clearance for that study. But there are certain aspects like the use of masks, the virus detection, or obviously lowering down the viral load in many cases has been proved really helpful. And I'm sure that you must have heard about an mRNA vaccine that has been developed with the help of nanotech, and it is quite the top of the town. So yeah, going about all this, we can also prevent the viral infection. We can improve the immune system response, obviously, with the help of nanofilms like PVA or polyolefin, with the help of nanocomposite materials, and also even certain gold and silver nanoparticles. We are basically doing a concoction of all sorts of everything that we have at hand in order to come up with a better product which will be cheaper and which will be better to use. I don't know if you have heard of that, but also in our lab we have almost 20-25% who are we go for the COVID test. We take the swabs. We do everything that's related to COVID on this hospital. So many of the people have generated rashes. Many of the people have generated face certain skin disease and all that due to the constant wearing of the PPE. And also we are trying to formulate how we can develop the material with the help of nanotechnology so that we can come up with a better product. Now, nanotechnology in all offers, we have got biogenetic nanofuels. We have got nanocomposite materials like I mentioned that we even have electronic nanotech films with the help of which we can at least, if not stop, we can reduce the amount of viral proliferation and the viral passage through those intercalations. So we are working on it. So it's a work in progress. The areas of ongoing research, as I have mentioned, these are all the areas: the drug delivery with the help of nanoparticles, reduction of the viral loads, therapeutic approach, and I mean the clinical things, the methods of newly formed drug delivery, faster drug delivery, and faster drug application and formulation. Everything is coming at a club, and we are trying to engineer every aspect of nanotechnology with the help of immunotech. So it's as I said, it's a mixture of immunoinformatics, which comes from the parent of bioinformatics, obviously. Now we are trying to go about doing every kind of possible resources at hand and giving a better way to it. So I want to cite this example. Dr. Pan et al. They had done a, it's being commercially developed by Vitruvian Bio, and it's a very recent study. They did a naked eye detection of COVID-19 with the help of a nanoparticle. Now they took the swab sample, the nasopharyngeal and oropharyngeal swab samples from patients, and they basically treated it with a visual color changing. It's just a colorimetric assay, and but there's the typical type of gold nanoparticles that they have generated, and this multivariate gold nanoparticles will give a dark blue coloration, and this will take for about 30 seconds to detect if you have a COVID-19, if you're infected with COVID-19. And the most important fact is this test has got almost 98% specificity and 95% of positive rates, and they haven't yet reported any false positive cases. So this is a way to look forward to nanotechnology at this point of time. This is how the nanotech started. Like we started working with nanotech. This table was primarily the motivation for us. This is the study that we have taken from the Journal of English Medicine. So this journal states that the SARS-CoV-2 virus in their PCID50, I mean the titer value, at this stage, the aerosol, the copper, the cardboard, the stainless steel, and the plastic, they took these 5 environments and they gave up, 10 results with respect to SARS-CoV-1 and SARS-CoV-2. But it was seen that in aerosol, almost along the time of the experiment, the viral particle was kind of, it was quite active in its state. But in case of copper, as you can see, the curve, the Bayesian, I mean, relation model shows that this curve is quite diminishing at a very fast rate. So it was just suggested with the help of this graph that how much COVID-19 viral particle might sustain on these various surfaces. So yeah, you can see me wearing the PPE that we are working on basically. So the PPE and the nanomaterials, yeah. So this PPE is kind of the market version of what we are getting, what we are using nowadays. But we are trying to make another layer in between this PPEs where we'll go about to combat the spread of coronavirus, and we are trying to increase the hydrophobicity, and also we are trying to introduce a kind of self-replenishing material where we, the surface plasmon resonance and all that factor won't give much of a importance, and the virus will die down soon. So the hydrophobicity and obviously the antimicrobial activity. Also, we know that certain nanomaterials have been infused with the help of sanitizers and all to create surface disinfectants and all that. So we are kind of working on this so that we can make this just a better project. And we have also applied to ICMR, but we have got the ethical clearance, and we are we are currently being funded by PHH and also by the NCI for our studies. So while doing this study, as you know, as I told you, I'm just a UG researcher and I don't have much of the resources at my hand. So I was thinking about how could I infuse, the problems that are coming in with everybody who's working around me, because as you know, the environment of working in live COVID situation, it's completely new to me. I have been to 3-4 labs, and that's like the part of my internships and all that. I have never been to a proper research facility for proper research before. So this is kind of like, yeah, I have done the internships and all, but this was the first proper exposure of mine to a research facility. So I was thinking that how can I introduce a research which will also give way to how people are feeling in this way. So I was very fortunate to get in touch with our clinical director, Dr. Bhumik, and also I got in touch with eminent psychiatrist of Kolkata, Dr. Bapajitto Chakraborty, and we have certainly come up with a new study where we are doing this, the COVID-19 mental health well-being studies, and we are trying to infuse these two studies as a control to our nanotech. Now, what we plan exactly to do is we are generating this type of PPE. We are working on this COVID-19 mental health study. Both are going at par, and both are already been funded and accepted by the ethical board. So we are not having any problem with that. So we are basically trying to take more considerations into the personal protection of the healthcare workers. The long hours at the front line, it's quite frustrating. Trust me, like when you are 8 hours, 9 hours without the food, without everything working, so it's quite frustrating for anybody out there. So the infrastructure is also somewhat, we are a developing country. As I said, we are working with our full strength. We are working to do whatever we can, but the infrastructure sometimes poses a kind of challenge to us. So we are trying to cope up to this new normal situation, as we all say. So as I said, that we are trying to get an overview that why not we take up a study, and we study this, and somewhere down the line we might introduce a control, we might introduce a better protection, a better protection formulation where we can predict that at least taking such effects would help us. So we have started a very interesting study together. So we are basically rolling out Google Forms, and we have all the informed consent and everything. So till date, we have come up with around 110 responses from COVID-19 healthcare professionals. And not to mention that in India, you will find, a lot of papers on every psychological survey of the doctors. But as Sir was saying in his previous talk, that the other sectors of these health and medicine and this research, everything, it's getting kind of neglected. So we started a study where we'll take the psychological views with respect to general anxiety disorder, and also somewhat down the line the dyslexia, and also we'll incorporate it with general depression and all that. So we were trying to collaborate and bring all of the things into a platter. So we thought of studying the psychological survey with the help of Dr. Chakraborty. We started studying the COVID-19 healthcare professional psychological mental well-being. So we rolled out forms, and we are also collecting responses as of now. So this is how we are going about, and I would like to share some of the very interesting observations that we have found, that this somewhere down the line, even in this situation when in India you hear a great deal of hopes in the name of news, and also there's like the pandemic is in a near state of fear. So at this point of time, somewhere down the line, we can see there's some hope and some silver lining that's coming up. So I am not actually allowed to share the major part of our research, because as it's ICMR protected and all that. But I would share some of the very futile details, but very interesting details that we have come through. So this was basically the part of the study, and we started taking the responses, and we had this question that have you ever been tested positive for COVID-19? And mind you, these are all the healthcare professionals, and this healthcare professional pool includes the doctors, obviously, the doctors, the nurses, everybody, those who are working at the hospital and nearby. And this has been collected pan India, not only Fearless or Eastern India. We have collected the data even from Mumbai, Delhi, and every part of the country. So they were kind of hesitant at first to give away the results. So we introduced this section that I have been tested, but I won't reveal my report. And you can see that most of the, like about 63% of the pool have not been tested positive for COVID-19. So we can at least hypothesize that the protection, the personal protection or the cure that we are taking, the healthcare workers are taking, are quite okay. But however, we are just trying to make it more economical and more resourceful. So this is one of the question, and the other one was that I am trained to take adequate infection protection, and most of them have said yes, which was quite to our surprise, because in my lab only I could see that out of 25 professionals that have been working in the lab only, those who don't get direct contact with COVID patients, about 10 or 12 or 10
sorry to interrupt, Radur sir. Actually, we have a time constraint of 15 minutes. Actually, it's going to end. If you can.
Yeah. Okay. Yeah. Okay. So I'm just ending. Okay. So yeah, from COVID-19 frontline workers, we have also got some interesting results that do you get restless or fidgety, and we have got this result. It's right in front of you. You can see how we are going to analyze, and all that's coming later. But we have also come up with another unique psychological factor, which has stated that are you struggling to stay motivated about your study? And as you know, going into the field every day, so you also have this result in front of you. So 20% of them are saying they agree that they are struggling. So somewhere down the line, our research is contributing to a faster implication of COVID-19 situation, and so that we can make it better for not only the healthcare workers but also for the people of the country in general. So our strength is basically this thing that many of the workers generally believe that yeah, whatever we are doing, it's basically giving some output, and I believe that we can achieve our goals faster and better with my contribution. So everybody of us, everyone of the country, the scientific community, the research community, the doctors, the healthcare professionals, everybody is going at large together, and I think this workforce and whatever we are doing is really commendable on our part. So this is the lab that where we work, and we do the testings and all here. We also, to introduce technology, we have we have come up with automated RNA extraction techniques by, I guess it's by Lipo Sigma and all that. So we are introducing new technologies where RT-PCR can be fast like made faster for the patients. We are testing about 250 to 300 patients a day. So all that is going on. So I'll try to acknowledge my guide, Dr. Shubhavati Bhomik, who is the clinical director of Fearless Hospital, Kolkata. I'll also like to mention about Dr. Bapal Chaudhary, who is basically taking up the effort to work with me on this psychiatric test and everything. So we have also members from Microbiology Society of India. We are we are also working with Dr. Shomishra Rai from Saha Institute of Nuclear Physics, where we are basically working on this PPE and all that. So we are trying to develop everything that we can do. So this is my email. You can reach out to me if you want. And thanks a lot. Thanks. It was great. Thank you.
Yeah, thank you so much, sir. Thank you so much, Raju sir, for the informative talk. So yes, so that is done. So thank you so much, all the speakers for today's session. It was really great having, because it was such a diverse range of topics. Right from stem cells to molecular medicine, and then going on till COVID-19. It was really great. So now I would like to have Ria take over the session. Thank you.
Thank you, Aditya sir, for beautifully moderating and all the eminent speakers for delivering such knowledgeable and interesting talks. We had a group discussion round with us among the students, but due to the time limit, we will shift it for tomorrow. The update schedule will be given. I would now like to invite Miss Mohini Agarwal for the vote of thanks. She is the director of JAX Foundation and ICMR Senior Research Fellow.
Thank you, Ria. I, on behalf of National Summit 2020 and the entire organizing team of JAX Foundation, extend a very hearty vote of thanks to all the dignitaries. I would like to thank our keynote speaker, Dr. Satyendra K. Rajput, Head of Pharmaceutical Department, Gurukul Kangri Vishwavidyalaya, Haridwar, for joining us and enlightening us with your words of wisdom. Our today's chief guest, Dr. W. Selvamurthy, President, Amity Science, Technology and Innovative Foundation, Director General for Amity Directorate of Science and Innovation and Chair Professor for Life Sciences. Shri Raghuraj Singh, State Minister, Labour and Employment, Uttar Pradesh. Dr. Harshvardhan, Minister of Health and Family Welfare, Government of India. Shri Satyendra Kumar Jain, Cabinet Minister, Government of Delhi. Dr. Ashwit Kumar Chaubey, Minister of Health and Family Welfare, Government of India. Some of them were not able to join us today due to some other engagements, but we require their blessings upon us always. Our guest of honor, Dr. Vinay Kumar Rail, Director, Symbiosis School of Biological Sciences, Pune. So thank you so much for taking out your valuable time and sharing your wonderful thoughts with us. I would like to thank all the eminent speakers for gracing their important work and sharing with us their findings and opinions. Dr. Jyotirmoy H., Professor, D.Y. Patil College, Mumbai. Ma'am spoke about drug repurposing in oncology. She has told us importance of drug repurposing in cancer and many other diseases. Dr. Shashi Kumar S., scientist at Avesthagen Private Limited, Bangalore. He talked about nutraceuticals and functional food for 20th century for alleviating lifestyle diseases like obesity, diabetes, and cardiovascular diseases, which is number one in mortality rates today. Dr. Shilpi Sharma, Professor, Savitribai Phule, Pune University. She spoke about HIAPP, a therapeutic target for type 2 diabetes. Dr. Praveen D. Podar, HOD Molecular Medicine and Stem Cell Biology, Mumbai. So has told about application of biotechnology in medicine, which is important practical application of biotechnology. And it's very important for our student participants, especially the upcoming biotechnologists. Dr. Preeti Chunarkar, Professor, Vidyapeeth Team University, Pune. Ma'am told us about data analysis and data center, which is important in today's world, and it is linked with all our experiments. Without without it, I don't think so. Any experiment would be complete. Dr. Swaroop K. Chakrabarti, co-founder, director, CEO, STEM Bionics Private Limited, Kolkata. He talked about cell therapy and precision management of human diseases. Mr. Radur Sarnadar, UG Research Assistant, Kolkata, who talked about nanobiotechnology and its application in SARS-CoV-2, the most important topic nowadays to eliminate this pandemic. I would also like to thank our today's bright and young moderator, Mr. Aditya Acharya, Executive Member of JAX Foundation, for beautifully moderating the session, and to all our lovely participants and academicians present here. Also, I would like to give hearty thanks to Director of Symbiosis School of Biological Sciences, Dr. Vinay Kumarel, who was a collaborator in today's summit, to Mr. Jitendra Kumar, Director and Founder of JAX Foundation, for perfectly managing his entire team, all the Board of Directors, Professor Kumud Bala, JAX Advisor, under whose guidance this foundation has started, Ms. Tanya Bhagat, HR Coordinator and Technical Head, and the entire team coordinators who made this event successful. Thank you, everyone. Over to you, Ria.
As the summit ends, I would like to invite you all to the day 2 of National Summit tomorrow at 10 a.m. and the links will be given to you. So it will be it would be appreciated if you join us tomorrow also. Thank you all again. Take care and enjoy.